Hello. Hello, Praveen. Yes, sir. Working, sir. Uh,
Sir.
Very good morning, all.
Nah,
Hello.
nah, can't ఇది బెటర్ కదా కార్డ్లెస్
Hello.
అన్నా అది ఇచ్చేసేసి ఇది ఇచ్చేసేయి.
Very good morning.
ఆఫ్ చేసేయండి.
Convener Emmanuel Kumar, Dr. Emmanuel Kumar, co-convener Anil Kumar, coordinator Ravi Kumar, and my friends and my students of this college and other college, and all the participants. Very good morning to you all. I am Dr. Anand from Government Degree College, Maheshwaram, now working at Women's College, Nalgonda. I am here to present a topic that is my work for my PhD. Actually, it is light switch on and off of ruthenium polypyridyl complex. Ruthenium, as we know that ruthenium is belong to the same group as that of iron. Iron, ruthenium, osmium. Iron is very biologically highly active and most important, having wide applications biologically. Similar to that, the same group, the other, that is, it belongs to 3D, whereas the 4D element is ruthenium. That is iron followed by ruthenium is there. Ruthenium also properties of ruthenium are also almost similar. That is why nowadays most of the research is concentrated or focused on ruthenium based drugs or drug carriers. Sometimes what happens means you require, there is a tumor in our body, the drug had to go exactly to the tumor. For that sake, they are used as carts, bullock cart, like that. Ruthenium carts are also used. Okay, that is, they are acting as drug carriers. Okay, for my the basic point is, in my this work, I had prepared a ligand which is novel. Ligand is there. This ligand is complex with ruthenium, and this ruthenium is having different types of ligand. That is two. One is called as intercalatory ligand. Another one is ancillary ligand. First of all, in our work, we had to prepare this one. That is phendione is prepared. Phendione, we had to take 110 phenanthrolene, in which concentrated sulfuric acid and nitric acid solvents are used, and it is reflexed followed by that is it is neutralized. Why? Because we are acidifying it. Therefore, we have to neutralize with sodium bicarbonate and extracted with dichloromethane. Is used, and finally we get the crystals of 110 phenanthrolene 5,6-dione. This is the starting material for my ligand. As I told you in the beginning itself, there are two types of ligand. One is ancillary ligand is there, another one is intercalatory ligand. Okay, we will know what is intercalatory ligand later. Later on, using this phendione, we have to couple them. Couple them using a compound that is methoxy that is 4-methoxy, 3-methoxy, 4-benzyloxy benzaldehyde. Okay, this both are coupled in the presence of ammonium acetate and acetic acid, and they are reflected to get this ligand. This ligand is purified later analyzed spectrometrically. It is analyzed spectroscopically. It was analyzed by UV, IR, NMR like that. Okay, in that way we are getting imidazole ring is there. Okay, this ring is nothing but imidazole ring. Okay, this is having wide biological importance, and this ligand is prepared. It is novel ligand. Then it is we have taken again. We have prepared this complex with phenanthrolene. Okay, that is taken and it okay, this complex is prepared, and this complex now this chlorine, and it is a huge macromolecule. A complex is prepared like this. We have prepared using this ligand. We have taken phenanthrolene as one of the ancillary ligand, and bipyridine as another one, and that is the hydrogen dipyridyl amine is also taken, and dimethyl bipyridine. These are the ancillary ligands, whereas this one is called as intercalatory ligand. Okay, why it is called as intercalatory ligand? Because for my work I am using DNA. DNA, as we know, is having helical structure. Okay, what I want to prove is this drug complex is having cytotoxic property. It is having anti-cancer property. For that sake, I am taking the work that is they have to prove that it breaks the DNA strands. The two strands that are there, it should break them. Then only it is proved. Therefore, based on the different studies, okay, one of the study which I used is a very simple one, that is UV absorption spectroscopy. In this one, if you observe here, we are having different peaks are obtained. These peaks, these two are with respect to pi to pi star transition. Okay, and this one is nothing but MLCT band, metal ligand charge transfer band. If you observe, the peak is slowly shifting towards the bathochromic shift. That is towards the red shift. It is obtained. That is when you add with the DNA, we are taking the complex and adding the DNA to this one. Okay, in increment level, slowly increasing the concentration of DNA. As the concentration of DNA is increased, okay, clearly it shows there is a shift in the absorption. Okay, this shift in the absorption shows that it clearly shows that it is having cytotoxic property. That is, it is cleaved. That is what there are three humps or three peaks in this one. Okay, these two are with respect to pi to pi star transitions, and this band especially is nearly 460 nanometers. At that range, this is with respect to the MLCT band. Okay, metal ligand charge transfer is going on there. Okay, by using this one, we can calculate Kb values, binding constant values also can be calculated. The second study which we did is fluorescence spectroscopic studies also we did. Okay, we have taken the complex and slowly increased the concentration of the DNA. As the concentration of the DNA increase, the fluorescence also is increasing. It is clearly shown that at nearly 620 nanometers, this is observed. Okay, this is again shows that the DNA is our complex is getting intercalated, is entering into the grooves of the DNA. There are many grooves. If you observe the DNA, it is a helical structure. There is a minor groove is there and the major groove is also there. If it enters that, our almost our complex is like a planar complex. Why? Because the ancillary ligand are phenanthrolene and our that is intercalating ligand, that ligand which interacts between the two strands, the hydrogen bonding is breaking there. Instead of hydrogen bonding, our complex will enter into that. Okay, then based on the UV absorption and fluorescence, the binding constant almost are correlated and their stability is also obtained. Fluorescence benching studies also we did. Okay, fluorescence benching studies also we did. Even in this one also, we can calculate the Stern-Wolmer constant also is calculated. Okay, fluorescence benching in this one, ethidium bromide is acting as a quencher. Okay, and actually the topic is light switch on and off. If you observe this one, if you take only the complex alone, this is the peak. When you add a complex with the DNA, the fluorescence is more. The luminescence or fluorescence is observed. But when you add, instead of DNA, along with that one, we add cobalt. There is a light switch off. Why? Because it is coming down. The fluorescence is decreased. That is in the beginning, the light is on. Whereas at the end, when you add cobalt, light is off. Why? Because here the DNA is almost free here. Complex and DNA is binded with cobalt. Now what happens is, if you add again EDTA, cobalt will bind with EDTA rather than DNA. Okay, therefore, again it is slowly increasing. This experiment we coined it as, our professor coined it as light switch on and off. Okay, there is an increase in the fluorescence as well as decrease in the fluorescence. Okay, if you want to switch on, we use a switch. Whereas here we have to add EDTA or add cobalt to switch on and off. Okay, so thank you all the organizers for giving me this opportunity. Thank you very much. Any question?
Your presentation is very nice, sir. I have one question. Already you have synthesized ruthenium complex and coupled with one more ligand, right? So you have prepared a bulk molecule. The activities may be increased or decreases compared to the already prepared ruthenium complex.
Both on our intercalating ligand, novelty is also required.
Okay.
Generally, use also there is an activity there, but
Already
it is a
ruthenium-
reported one.
Ruthenium, phenanthrene, chlorine compound is already reported.
But its activity is very less.
Okay. After coupled with your ligand, it has been enhanced.
Enhanced by the intercalating ligand, especially ligand index. It is planar and planar is also proved by extractive technique.
What is the complex structure, sir? After your synthesis?
After
head.
I have couples of questions. I think it's your presentation question. How your presentation is related to this webinar seminar?
Anti-cancer properties. The product area, like that ancillary area.
Topic is sustainable development, right? How we have to connect somehow. Our contain anything, we have to connect somehow. Another thing, you have
Sustainable development goals. Here, consequences of this fact, when compared to this fact will have more consequences. Side effects are more. Whereas this for ruthenium almost similar to that of iron, and it's forming this pattern is a spare planar complex. Whereas this one is octahedral, almost similar to our
another question is that how this is the carrier. You mentioned that this your drug is working as a carrier.
No, sir, my project is not that.
No,
What I
in the
introduced,
presentation you have mentioned
that ruthenium is nowadays are used as gauge comp, cathode comp. Where ruthenium will be the metal, it will be there, it will hold the gauge. and the drug is inserted into that cage, and this drug goes to the tumor site where the pH will be very less. At that site, it gets disintegrated. That is why it is acting as a CAR.
What is the difference between cytotoxicity and anti-cancer? You have mentioned that your compound is showing that anti-cytotoxicity as well as anti-cancer.
Sir, here cytotoxicity means if you just take a cell, tissue is there.
No, I'm asking what is the difference?
It kills the cells.
Yeah.
Cytotoxic means it is toxic for a cell.
The normal cells or tumor cells or any cell?
Generally, it is
Okay. Cytotoxicity means normal cell. It should not have the cytotoxicity. It should be have the anti-cancer. You have to mention that. Okay, that you have to correct it. Another thing is that you also have mentioned the UV pi transition two peaks. It won't be the two peaks for the pi transition. You have to correct that one also. And the PL intensity of the third figure and fourth figure are quite different.
fluorescent techniques are different.
It should not show that in the presentation or anyway. It should be the uniformity. Okay, na? And also here the mechanism is not clear why it is the EDTA, why it's not DNA. We have to explain. We don't have much time. I'm just giving the inputs. The interaction mechanisms you should have to make it clear. Your content also you have to make it clear. So this is just suggestion for you. Okay. Overall the presentation is good.
Thank you very much, sir. Your presentation is very nice, and the compounds you have prepared with ruthenium metal is nice. My question is that during the intercalating process, the ruthenium complex, is it intercalating with the chemical bonds or physical forces with DNA?
Just biophysical process.
Hydrogen bondings.
Okay. Physically, it enters into that area and breaks the bond.
Okay. Is there any apoptosis process during the drug action?
Our ruthenium compound properties also proved by different techniques like cyclovoltammetry studies also proved the apoptosis. Our analysis and we work with them. And Ravi sir is sent on it. Thank you. This is the result. Yeah.
Sir, thank you, Dayanand sir, for coming here and presenting your work.
Morning, respective judges on the dais and convener, co-convener of the seminar, and dear students, participants in the seminar. I'm R.B. Ramchandar from Tara Government College, Assistant Professor of Microbiology. Today I'm presenting on a study on endophytic fungi in peanut seeds of Telangana state and biocontrol of seed-borne endophytic fungi. Peanuts, we know groundnut, it is vulnerable to number of fungi because it is there in the soil. It is susceptible to number of fungi. Peanut grains which are stored, they contain number of fungi. Researchers earlier isolated Aspergillus species, Aspergillus niger, Aspergillus flavus, Alternaria, Fusarium, and number of pathogenic fungi also, amongst which Aspergillus species, Aspergillus and Fumonisins that was produced by Fusarium, aflatoxins, these are poisonous metabolites produced by Aspergillus parasiticus and Aspergillus flavus. Fumonisins, these are produced by Fusarium species.
These are responsible for cancer, colon cancer, liver damage, liver cancer, particularly aflatoxins. And fumonisins, they are involved in esophageal cancer. Remaining are also fungal toxins, mycotoxins. They are also having some effects such as vomiting, blurred vision, hypotension, etc. Amongst which aflatoxins and fumonisins, those are more poisonous. In very high amounts, they may cause also death of the individuals who consumed these foods. Not only peanuts, groundnut, there are several other foods such as maize and certain other foods also involved in aflatoxin contamination. Several foods, they contain these fungi, aflatoxins. Consumption of such food leads to this liver cancer and other consequences. This present study is aimed to identify which fungi are present in peanuts of different regions of Telangana state. What are the different endophytic fungi are present in the peanuts collected from different districts of Telangana state. And the study is also aimed to investigate antifungal potential of leaf extracts of Tamarindus indica and Adatoda vasica. Tamarindus indica, generally we use tamarind in our foods, and which was known as antifungal. Adatoda vasica is a medicinal plant which is having some antifungal effect. Isolation of endophytic fungi from peanut seeds collected from various regions of Telangana, and isolation of these fungi, identification by morphology and microscopy up to the general level, and evaluation of phytoextracts for biocontrol of these fungi, and preliminary phytochemical analysis with existing laboratory infrastructure in degree colleges. The methodology, my students helped me to collect the seeds from different districts of Telangana, from different areas, Jogipet, Jairabad, Gajwel, Bachpalli, Sangareddy, Mahbubnagar, Nagar Karnool and Jadcherla. We collected the seeds, and these peanut seeds are sterilized first. The surface, we are going to endophytic fungi inside which are there. Just the peanut seeds are placed in 70% ethanol for few minutes. Then they are cleaned by placing in distilled water. Then the peanuts are inoculated into, they are placed in potato dextrose agar medium, which is a natural medium for isolation of fungi. And the number of fungal colonies which are there surrounding the seed, they are counted. Microscopy was normally used, microscope was used to identify the fungal isolates which are there, and the fungi were placed in the lactophenol cotton blue on this slide, and the fungal genera are identified based on colony morphology and microscopy. Fungal genera were identified, which fungi are present. Then the leaves are collected, Adatoda vasica and Tamarindus indica leaves. Leaves are washed and extract is prepared. 70% ethanol is used to extract the extracts, and these suspensions are dried in oven. Powder form it is made. And antifungal activity of these extracts was tested by Poison food method. Potato dextrose agar medium is prepared after autoclaving at cooling at 45 degrees. The extract powder is added in the concentration of 1 mg per ml. Then medium is poured into Petri plates. After that, this fungi which we isolated, which was grown, that is taken 5 mm disc. 5 mm disc was taken with steel borer, and we placed that disc on the medium. And control plates without extract also taken. And we also carried phytochemical analysis by using normal preliminary phytochemical analysis to detect the secondary metabolites which are there in the extract. Wagner test for alkalides and test for indole alkalides using PDAB, and test for tannins, and test for saponins, protein test for terpenoids, and we also did for quinones, coumarins,
using fluorescence, yellow fluorescence on UV trans illuminator, and resins, test for resins, test for flavonoids, and these are the results. These are the fungi, these are the peanut seeds which are placed on medium showing the fungal growth. Uh, these are the fungi which we isolated: Aspergillus, Rhizopus, Geotrichum, Mucar, and Fusarium species were found in the seeds collected from different districts. In Jogipet, seeds collected from Jogipet, we exclusively got Rhizopus, almost all seeds. In Jahirabad, we got Mucar. In Gajwel, Aspergillus and Rhizopus. In Bachupalli, we got three species: Aspergillus, Fusarium, and Geotrichum. In Sangareddy, Aspergillus and Rhizopus. In Mahbubnagar, Aspergillus and Mucar. In Nagar Karnool, we have Aspergillus, Geotrichum, and Rhizopus. In Jadcherla, or exclusively seeds, almost all seeds have Aspergillus. This is the distribution pattern, and figure B, you can see occurrence of mycotoxigenic fungi, Aspergillus and Fusarium, in peanuts of different locations of Telangana. Aspergillus is known for production of aflatoxins, Fusarium for fumonisins. Whereas Jogipet, Jahirabad, these Aspergillus and Fusarium are absent, but in remaining areas in different percentages they are present. These are the results of poisonous food technique. Our extracts are showing antifungal activity. So against Aspergillus, Rhizopus, Mucar, Fusarium, these are tested. And percentage activity, you can see diameter. This is diameter. How control you can see how much it is, and in extract how much diameter, how much percent they are inhibited. Diameter of fungal colony in PDA plate with the extract of Adathoda vasica, and the diameter of fungal colony with Tamarindus indica. You can see the diameter. Control plates, they are extensive growth, whereas with extract inhibition of the fungi. These are the results showing. So control plate fully for fungal colony we can see, whereas with the extract the growth is completely inhibited.
invite Jyotsna Madam to forward the proceedings.
Continuing the proceedings, let us invite the dignitaries for this technical session. The resource person is Professor Gangaraju sir, the brain pool professor from Changyang University, to come onto the dais. Sir, please come onto the dais. Let us invite the chairperson of this session, Dr. Ravi Kumar sir, our vice principal. And the rapporteur for this session is Dr. Sitish Gaur sir, to come onto the dais. I request Ravi Kumar sir to continue the proceedings.
Good morning to all. This is Dr. Ravi Kumar Parangi, Associate Professor of Chemistry, NG College, Nalgonda. On behalf of two-day national seminar, which is conducted in NG College yesterday and today, I have great privilege to introduce Dr. Gangaraju Gidde. He is currently services as a brain pool professor at Changyang University, which is 11th rank in Korea, South Korea, and also worked as a associate professor at NITTE University, Mangalore, India. His innovative research focus on green synthesis of nanomaterials, bioimaging, targeted drug delivery, clinical biosensor, and environmental remediation, making sustainable contribution to the both biomedical innovations and advancement of sustainable practices. Dr. Gedde hold a PhD in biotechnology from National Sun Yat-sen University, Taiwan in 2010 to 15, where he specialized in the synthesis of characterization and biomedical applications of nanomaterials, nanoparticles. Prior to that, he has pursued Masters of Science in Analytical Chemistry from JNTU, Hyderabad in 2007 to 2009, specialized in electrospray ionization tendon mass spectrometry for peptide analysis. His contribution have been recognized through the prestigious awards and fellowships, including Brain Pool Fellowship, South Korea, from 23 to 25, and the most postdoctoral fellowship, Taiwan, from 2015 to 17. He secured research funding from Department of Science and Technology, DST, and Department of Biotechnology, DBT, in India in the capacity of principal investigator. As active member of the global scientific community, Dr. Gedde serves as a editor and peer reviewer for leading journals. He is regularly invited to deliver and keynote lectures at international conferences with over 40 peer-reviewed publications and 10 book chapters. Total citation 2319 and H index is 24. And highest impact factor is 19. The finding of his research have garnered premiances of international level. He had a participate in several funded research projects, collaborative partnership with institutions in South Korea, Taiwan, Saudi Arabia, Ethiopia, Indonesia, Poland, and Malaysia to the commitment to scientific excellence and global collaboration. Thank you, sir, and thank you very much. Please carry on.
మీరు ఆఫ్ చేస్తే నేను స్టార్ట్ చేస్తాను, లేదంటే స్టార్ట్ చేయండి. మన ఇద్దరం మొదలు పెడదాం. ఓకే, I request all the students focus on the presentation. Yeah, నేను కూడా అలా చేశా. Okay. As the sir discussed about my biography, most of the information I know people didn't understand the student level. Okay, it's fine, but try to focus on the content. Okay, so can you start? File name, sir. File name. I think NT college. Yes. Actually, when they invited me as the talk on this seminar, I was a little bit confused because I am the nanotechnology guy. But the presentation is that life science and sustainable development goals. It takes some time to connect my research work towards the sustainable development goals. So here I have picked up the one topic, that is the plant leaves. So and I connected with the nanotechnology and how it is sustainable. Okay. The title is Sustainable Biomedical Innovation through Nature Inspired Nanomedicine. Okay. So it means that we are developing the nanomedicine from the nature and it should be sustainable. So let us see the outline, what we are going to discuss. First thing is sustainable development goals. I think yesterday many speakers, they speak about the sustainable development goals. Can anyone tell how many goals? Even I am not related to these goals. How many sustainability goals, SDS? I think yesterday you people attended the sections, right? Okay. I will discuss that one. Next, nanomedicine, biomedical application, these two things. Another thing, the most important, I think the students here, degree or PG or PhD, I am going to discuss about the international opportunities, which are the fully funded, which are more important to you students, I guess. So these are the three sections I will cover. First one is that sustainable development goals. I will make it brief. Next section, I will discuss about the nanomedicine from the nature. Finally, I will discuss about the international internships. It means that 3 months, 6 months you can go and work, and also internships. Those internships you don't need to pay any money. How we can go from the even the degree college also, how we can go to the abroad or some IITs, NITs, ISRO. The base I will also discuss on the last section. So the total, the sustainable development goals 17. Okay, you people can search on it. I'm not going in the details. In that, I have selected one goal, that is the third goal. Sustainable development goal third, that is ensure health, lives, and promote the well-being for the all. So health, primarily I am going to discuss today health, and it is related to the life science. Okay, life science means I am going for the plants. Plants, how it is help to the how it is connected to the nanotechnology and how it is help to the health. That is the things I am going to connect and give the explanation all of you. So before going to nanotechnology, we generally heard about the nanotechnology anyone, but there is a confusion. Lot of the things. Why this nanotechnology is so important? Right nowadays, present days. The most exciting technology is AI. But
Nano
Dude,
also still
sir.
growing. But
Dude.
why? Anything when you are learning, the fundamental is very important. If you understand this slide, the nanotechnology, everything you will understand very clearly. So just 2 minutes, if you focus on it, you can understand. So the fundamental principle of the nanotechnology means there is two words. One is nano, another one is technology. Okay, nano means very small, extremely small. Okay? And why this small nano is so important? This is the question. Why this small nano is so important? How it is plays a role and how it is important in the many applications. If you take a lot of any application, biology, chemistry, mathematics, engineering, biotechnology, everyone nowadays applying the nanotechnology. But the word is very nano. It is from also the material. But why it is so important? That's the point. This is the fundamental principle of the nanotechnology. Nano means small size. Size is decreasing. We are decreasing the size. When you decrease the size, the surface area is increasing. When you decrease the size, surface area is increasing. This is only the fundamental things in the nanotechnology. This is only the one thing in the nanotechnology, the fundamental principle. The size is decreasing, surface area is increasing. Anyone can explain this one? The size decreases, how it is increasing surface? It should be decreased, right? Small size have more surface or large size has more surface? Which one have more surface? మీలో మీరు ముచ్చట్లు పెట్టుకుంటే కంటెంట్ అర్థం కాదు. Then it's no meaning I came from the Korea to here. Okay, try to understand. It's very important in your future also. Even I'm more PGS person. Okay, I didn't have time because of degree college people don't get the opportunities like me. That's why I came here. Okay, if I go to the universities, the facilities are much higher than here, but I came here because of you. If you are making nonsense, why I have to be? I will go out. I am also came from here. Okay, my father is farmer, my mother is farmer. They don't know education. I also work in the farm. Okay, I came only because of you, not from the university college. If you don't listen here, go out. Why I'm here? I came only because of you, not for the college. Because government college students don't have the opportunities. They don't have the exposure. We have a lot of the opportunities. Okay, my father no education, my mother no education. My brother is IIT Kanpur. He is working in the leading international company. I am world top 100 scientist. My younger brother also completed PhD. My mother and father no education. We are in the rural area, small village. Okay, I came because of you. Try to focus on here. Okay, let us see how size is decreasing, surface area is increasing. If you take this part, okay, what is the surface area here? 6 centimeters. If we break it, there are several parts, right? If you break it, there is several parts. I will explain more clearly. If you take this stone, this is the surface area, this is the surface area. Calculate. When we break it here, there is also 2 more surface area. You have to calculate total surface area. If you break again, the surface area again increases. So you have to calculate total surface area. So here we have broken it. All the part surface area, when you calculate it, it is a 24 centimeters. It is increasing from 6 centimeters to 24 centimeters. So size is decreasing, surface area is increasing. If you break again, the surface area is again increasing. So it is 48 centimeters. When you are decreasing size, surface area increasing, the optical properties, fluorescence properties, magnetic properties, electronic properties, all the properties will increase. Okay, that's the reason the nano is more
useful. I will give you one example. In our daily life in home, we will use the filters, right? The filters, the black color we will see, that is the carbon. Okay? Why we are using the carbon? It will absorb the contamination. Mostly the metal contamination or microorganisms, they will absorb, right? It is again that depends on the surface area. Okay? If the surface area is more, they will absorb more. If the surface area is less, they will absorb less. So if I take 1 gram bulk means nano color. It's not nano, okay? Bulk or macro. Bulk, we will say generally bulk molecules. So I take the bulk 1 gram or 1 kg. At the same time, I take 1 kg nano, which is have more surface. Nano have more surface or bulk have more surface? Nano have more surface. When we have more surface on nano, they will absorb more contaminations, right? So the purity is nano is more than bulk, right? All the application it is happening. So size is under Japali. Everyone have to say when the size is decreased, surface area is increased. When the surface area is increasing, optical properties, magnetic properties, electronic properties, fluorescence properties, all the properties will enhance. That's only the fundamental things in the nanotechnology. So, in the nanomaterials, biomedical application, basically we treat therapy and diagnosis, treatments and diagnosis. Okay? In the treatment and diagnosis, there is another important thing, the targeting. We have to deliver our drugs target precisely. That's why when you are using the medicine, we will have the several side effects. So because the drugs is circulating in the blood, it is not reaching into the particular part. That's how we'll explain how it is reaching that one. So in the biomedical application, broadly we classify, one is treatments, another one is diagnosis. First we have to diagnose what is the disease. We have to identify it. That's I will go into discuss, and also I will also go into discuss of the treatments, how the precise treatment, target treatment will happen. I will discuss in the later sections. So there are the several plenty of the nanomaterials, polymer nanomaterials, dendrimers, and liposomes, solid lipid SLP, and quantum dots. There are several quantum dots like cadmium, CDS, CDT, PPS. There are varieties of quantum dots. Quantum dots literally generating the size less than 10 nanometers. They will show the fluorescence properties, quantum properties. That's why they call quantum dots. And silica nanoparticles. It is also like the carbon cycle. When you are studying the chemistry, the silica also similar to the carbon structure. And also gold. I have mentioned one gold, but it's the kind of the metals. There is a gold, silver, copper, many metals, nanomaterials. And also magnetic nanoparticles. Some of the materials they have the magnetic properties like iron oxide, Mn, NiO2. Those some of the nanomaterials they show the magnetic properties also. And also there is carbon nanotubes, graphene, carbon fibers, single wall carbon tubes, multi-wall carbon. There are many. So nanometers, there are plenty of varieties of nanomaterials. There is 2D nanometers, 1D nanometer, 3D nanometer, 0 dimension. There are many classifications. We don't need to go more details. So here the preparation. Here I'm going to discuss about the preparation. During the preparation of the nanomaterials, what is the role of life science? Generally, when we are preparing the nanomaterials, there is a charge Au3 plus, Ag2 plus. The charge we are going to make the 0. So when you are going to that one, we need a oxidation or reduction. Simple. Electron accepting, electron donating. So these things we generally use the oxidizing agents and the reducing agents. So the oxidizing agents or reducing agents, they will give the electrons or donating the electrons or taking the electrons. The plants also having the multiple phytochemicals. So those also can act as a electron donor or electron acceptor. So it means that the plants leaves can act as a reducing agents. And also when you are preparing the nanomaterials, there are several problems like aggregation. They will come together. When we are using the plant extraction, there is some phytochemicals. They will work as the protecting agents. So we can get the clear nanomaterials. Okay. So here we can see that first we have to take the plant extract, then you can use the metal salt, any metal, copper metal, silver metal, gold metal, AuCl4. You can also prepare these things in your chemistry simple lab. Okay. Then you have to use some temperature or pressure and 90 degrees, 180 degrees, and time and temperature. Then finally we can see the color change from starting to end. So then we can prepare. So here the nano green plant leaves can act as a reducing agent. Agents are protecting agents. They will protect the nanomaterials. So here the plant leaves as the part of the nanomaterial preparation. So the nanomaterials where they prepare, they have applied in the several applications: antifungal, antibacterial, antiviral, anti-cancer, and wound healing, and drug delivery, and also biosensor. So here the plants are used as a oxidizing agents or reducing agent to prepare the nanomaterials. Okay, when you are using the commercial oxidizing agents or reducing agents, we have purchased, and those are preparing in the laboratories, right? Those are preparing in the laboratories. They are not sustainable, but plants are sustainable, right? So when you are incorporating plants in the preparation of the nanomaterials, we can prepare forever. We can prepare forever. So this method is much better than chemical synthesis. That's why we call the name green synthesis. Green synthesis of nanomaterials. Because here we are avoiding the more chemicals for the preparation. As I mentioned initially, there are two things. One is the diagnosis and therapy. This I am going to discuss. We have used one plant to prepare the silver nanomaterials, and we use the dye degradation and also antibacterial properties. This is the overall process. We have collected the plants and make the powder and treat with the silver nanomaterials at the different temperatures. Okay, different time. We have to start from the 50 degrees to 120 degrees and 2 hours, 4 hours, 6 hours, and 12 hours like that. After formation of nanomaterials, we have to do several characterizations like UV, fluorescence, IR, XRD, TEM, XPX. There are several things to confirm the nanomaterial. Then we have performed the antibacterial and also dye degradation. And also there is another material from the nature. Okay, there is another material from the nature I am going to discuss. This is very interesting material and world leading material. Before that, the family of that material. The family of that material. The family is carbon materials family. Okay, the carbon material family, there are many carbon fullerenes, fibers, graphene, single wall, double wall, and there is a one material 2004 or 2005. Can you read anyone? That is the carbon dot. The carbon dots, they are investigated in the 2004. అంటే మనం పుట్టినప్పుడే నేను రీసెర్చ్ చేస్తున్నప్పుడు నేను కొత్తగా కనిపెట్టగలను అనుకున్నాను స్టార్టింగ్ లో. But this material has started in our generation, and it is the leading among the all the nanomaterials. Why it is leading, I will explain. And it's also from the nature. Graphene is not from the nature. Carbon nanotubes are not from the nature. Carbon fibers are not from the nature. But carbon dots we can produce from the nature. Means plants. You can see the carbon dots, the publications have started from the zero and increasing and increasing. It means that these materials are leading nanomaterials in the world. So the history, there is a small history about the carbon dots investigation. The Zhu et al., the scientists, Zhu et al., they are preparing the single wall nanotubes. While preparing single wall nanotubes, they are purifying by electrophoresis. When they are purifying, generally we will collect the precipitate. Generally we will collect precipitate and we will throw the supernatant. Solution part we will throw and the powder part we will take it. The solution part, when they throw, they unfortunately throw under the UV light. When they throw under the UV light, it is shown the blue color. The emission, it shows the blue color. Then there shows the interest how it is possible. Then they found that under the TEM, transmission electron microscope, the size is less than 10 nanometers. Generally, carbon materials more than 10 nanometers. This is very small and it is showing the fluorescence. Then they confirmed several techniques and they found that initially the carbon dot, because it is carbon in the shape of the dot, graphene is not in the shape of the dot. Single wall nanotubes like cubes, they are also not dot. So the carbon it is in the dot, that's why they named the carbon dots. There are several names like carbon nanodots, carbon dots, carbon quantum dots. Still they are calling carbon quantum dots, but it's not confirmed. It is showing the carbon conformity or not. Still the studies are going on it. So there are the two types of the methods to prepare the nanomaterials. One is the top down, another one is that bottom up. The carbon dots also we prepare from the either top down or bottom up. So these nanomaterials, as I mentioned, we can prepare from on the different fluorescence, green, blue, red, any color we can prepare. It is based on the precursor selection, temperature, and reaction conditions. And why we have to prepare these things and it is how it is related, I will explain in the next slides. So generally we need several techniques, as I mentioned, transmission electron microscope. It will show the size of the nanometer, size and shape of the nanometer, how much size is there, what is the shape, we can identify from that term. Another one is that UV visible absorption range, what is our material absorption range, we can find out. Another one is fluorescence spectroscopy. The fluorescence spectroscopy, we can find out the material is fluorescence or not. Another one is that IR. IR will tell surface functional groups, OH, NH, COH functional groups. What kind of functional groups exist on the material, we can find out from the IR. These are the fundamental techniques to confirm the carbon dot. There are several, as I mentioned, XTX, XRD, Raman, okay, there are many things we can use, but these techniques basically we require to confirm the material. Why this material is that synthesis is green and nano size? This
Carbon dot itself have the surface functional groups like OH, COH,
NH. If you take the gold don't have any surface functional groups. If you take the copper, no functional groups. But the carbon dots itself having the surface functional groups and they are biocompatible and means less toxic and they are water soluble. Most of the metal nanomaterials suffer with the water solubility. We can prepare very easily, but they are not soluble in the water. This is the one of the interest investigation. I have found it. This from the Nature. Nature is the topest. If you see the Nobel Prize, generally we heard after the Nobel Prize, these journals will lead Science and Nature, the top research journals. When I am reading this article 2010, during that time, I found that the worldwide there is a lot of seafood waste is there. In generally, we in home we use 1 kg or half kg, it's not more. But in the industry, the scale is very high. So when I see that is not soluble in the water, they are dumping in the sea or somewhere else, they are producing toxins. And dumping is economical problem. I have to think that what are the chemical components present in the sea shell? That is the seafood waste. What is sustainability? Don't waste seafood waste. Okay, it means that we can use out the seafood waste, the crab shell, prawn shell, even the fish, we can use it. How? So when I'm reading this article, there are two chemicals. One is that chitosan. What is that? Chitosan. Chitosan is the polymer. Another one mineral is there, that is the calcium. Okay, one is chitosan, another one is calcium. I thought that how these compounds can be useful. If I make some useful compounds from this material, it is sustainable, right? If I make some useful material from this waste,
it is the sustainable. So how I did it? I have taken the crab shell. Okay, I have taken the crab shell. The crab shell I already mentioned, it is having the polymer, that is the chitin. The chitin have the carbon. Okay, so then I use the ultrasonication. They will produce temperature and pressure, and I found that the blue color dots, nothing but carbon dots. So now I no need to waste the seafood waste. I prepared the carbon dots from the seafood waste. Because my father is farmer, I always think how to use the waste. Okay, then I have conjugated folic acid. The women, they generally they take the folic acid. So the folic acid will interact with the tumor cells. The folic acid will interact with the tumor cells because the tumor cells have the receptors. Tumor cell has different receptors. Normal cell have different receptors. This folic acid only interact with the tumor cell. Okay, this folic acid only interact with the tumor cells. Now my compound is fluorescence. I have tagged with the targeting agent. I have treated with the normal cells and also treated with the cancer cells. When I treated cancer cells, this compound cell attracted with the cells more. You can see the blue color and the cancer cells. So generally, the organic dyes they use for the imaging of the fluorescence. The organic dyes are very expensive. The synthesis is very complicated. The purification is more complicated. Our carbon dioxide came from crab shell. Is it expensive? The synthesis that simply ultrasonication, it is available in our labs also. For the suspension, we use the ultrasonication, right? So this method, the material already shown. This was published in the very good journals. Almost 10 publications I have published here. When I started this work, all my teams are laughed. Hey, this guy is going market and bringing the prawn shell, drying, the smell is coming. What nonsense you are doing? I've done Taiwan, right? Everyone is surprised and they don't understand what I'm doing. Okay? In general, when we do the research, we study some papers and we follow them that one. But when I'm doing that one, they're worried what this nonsense guy is doing every day. In the lab is always smell. But when the papers are published, they shock. So we also publish from the nest. Before I use the carbon dots from the crab shell, use it for the fluorescence imaging. Now what I did, additionally, I have incorporated magnetic elements. What I done? Incorporated magnetic elements. Mn, iron, gadolinium. Now what I have, one is fluorescence property I have, another one magnetic properties I have. Okay, generally we have the very serious problem. Okay, we go to the MRI imaging, right? When we are going to the MRI imaging, they use the MRI contrast agents. That is a gadolinium complex. The gadolinium complex are very toxic. Okay, the gadolinium compounds are very toxic. So what we did, we incorporate the gadolinium ion inside the carbon dots. When we incorporate inside, they don't show the toxic. So the crab shell used to prepare the carbon dots and magnetic carbon dots. Before the carbon dot is fluorescence carbon dot. Now carbon dots is magnetic fluorescence carbon dot. It can be used the fluorescence imaging and also magnetic imaging. We also incorporate the anti-cancer drug. That's I will discuss in the later. So it will be used for the anti-cancer treatment, fluorescence imaging, magnetic imaging. It is published in the ACS Applied Chemistry more than 10 impact factor. When I'm doing this people paper, they are laughing. But it's published in the very leading journal. So the down we can see the white color is increasing. When the concentration of my compound is increasing, the white color, if you see the XRT, there is a contrast black and white. Right? In the MRI also, there is a black and white. So here I already proved that the carbon dot from the crab shell, it is showing the
magnetic property, fluorescence. The seafood waste is sustainable. Okay, your goal is reaching my work, right? So when I'm doing this work, I have got very funny idea. When I'm doing this one, the chitosan I am extracting, right? I am extracting what is the compound? Chitosan compound. When I am extracting that compound, I am throwing calcium. I am treating with HCl. CaCl2 is going outside. While that I am reading some papers, there is calcium nanomaterial. I thought that why should I convert this one is calcium? I am throwing right anyway the solution. Okay, I found that very beautiful plates. It is a very unique material. Generally, we see the different shapes of the material. We never seen the plates. Okay, this work is published in the Green Chemistry. It is a dream to publish every chemistry person, this journal. Even when it is published, I didn't get the slip 2 days because I didn't expect my work will be go and published there. And we shown that antibacterial properties. So even the carbon dots, we can also prepare from the plant leaves. These plant leaves prepare carbon dots. We use it for the treatments and also diagnosis. Why the plants shows that they have the plenty of phytochemicals, terpenoids, polyphenols, vitamin C, okay, so on. So because of these phytochemicals, they shows that different bioactivities. So not only plants, leaves, we can also prepare the fruits, food waste, different, all the sources. These all sources are from the nature. So my carbon dots prepared from the nature, it's obviously sustainable. It's obviously sustainable. So when I prepare these carbon dots, okay, before going to the any treatment, anything, we have to study the biocompatibility. It should show less toxic. I have used a zebra fish from egg to fish. We shown that our material shows that very high biocompatible. It is all works are done in my laboratory. After that, I have used that carbon dioxide as mentioned before. Cancer cell imaging. It's maybe not clear. It is showing that blue, red, and orange. So the carbon dot shows that different cell imaging, multicolor imaging. We call it is the multicolor imaging. We also done zebrafish imaging also. So it means that the carbon dots came from the nature. They shows that very good bioimaging agents. It's a very good bioimaging. Imaging nothing but diagnosis. Imaging nothing but diagnosis. So the carbon dots from the nature we can use as the imaging agent or diagnosis. And another thing, now I'm going to discuss how these nanomaterials or carbon dots work for the drug delivery. I have mentioned that biomedical applications. One is the diagnosis, another one is the treatment. In the diagnosis, I already fulfilled the nature carbon dots are useful for the bioimaging agent. Now I am going to discuss how these carbon dots work as the drug delivery. So most of our commercial drugs have the water solubility and they won't reach the targeting. They don't reach target. That's why we have the different types of side effects. So the target is very important. Our drug should reach to the target place. It is very important. So what will happen? There are three kinds of molecules. The first one is that nanomaterial, and the arrow top, that is targeting agent. The down one, drug. So there are three things. One is nanomaterial, targeting agent, drug. I will give you a very simple example for here. Wait a minute. So we use the Zomato Swiggy, right? We generally use the Zomato Swiggy food for ordering the food. In Nalgonda, Zomato Swiggy. Madhyanam aithe em tinali em cheyali ఇది అంతే దిగులే జొమాటోలో చూస్తే అయిపోద్ది, చెట్టు కిందకి వెళ్తే అయిపోద్ది. అంతేనా? ఓకే, దీన్ని మనం అక్కడ కనెక్ట్ చేద్దాం. Zomato, when we order the food in the Zomato, the vehicle is there, right? You should have to use the vehicle, right? You should use the vehicle, right? That vehicle nothing but nanomaterial. Okay, well, the vehicle what he used, that is nothing but nanomaterial. So it means to carry the food. He used the GPS, right? When he is carrying the food, he used the GPS map. We will share the location or we will give the address. Then he used the GPS. It will be help to the target. The GPS will help to target. The target molecule will also help to reach to the tumor cell. Okay, the nanomaterial is vehicle, GPS is targeting agent. The food we need, what we order, the food, right? To fill our stomach. Their food is dogs for the treatment. Okay, so what is happening? The vehicle is carrying the food. The vehicle is carrying the food. Targeting agent reaching to the tumor, because I mentioned earlier the tumor have the specific receptor, which are the different from the normal cells. The cells in the tumor is different. The cells in the normal cells are different. So this targeting agent will help to the reach to the tumor. So nanomaterial and the targeting agent along with the drug, it is reached to the tumor. Okay, na? So now the drug is reached to the tumor. Now the main role is that the release of the drug. How it is released, right? When we are taking the medicine, the drug is circulating the body. My material also can circulate the body. That's why it is specifically the drug and everything reach the target. How it is released. Here what will happen? The body pH is neutral. Our body pH is neutral and the tumor pH is lower than the neutral, either 5 or 6. The bond between drug and nanomaterial, we will make it is stable at the pH 7, unstable at lower pH. So when this compound is reached to the tumor, because of the weakness of this bond, the drug will reach to the tumor cell. So that's the why the nanomaterial can work as the targeting drug delivery. The total word we call as targeting drug delivery. Now we will go how these carbon dots work as. Okay, now we will go how these carbon dots work as the delivery of drugs. In our work, we have prepared the blue color carbon dots and conjugated with the folic acid, and also we have incorporated the doxorubicin. The doxorubicin is the anti-cancer drug. We have found that in the lower pH, it shows that the anti-cancer property. We can see the drug release is very high in the lower pH. The blue color is the lower pH. Okay, so it means that our carbon dots from the nature successfully used for the delivery. Which delivery? Targeting drug delivery. The carbon dots from the nature successfully used for the targeting drug delivery. This is the confocal microscope image to see the drug really releasing or not. We also use green tea leaf. Every day in the Taiwan, we use that lots of the green tea. Okay, yeah, our lunch, breakfast, everything we use the green tea. When I'm drinking that tea, I'm throwing the tea waste. I thought, why can't use the tea waste? Then I have used the green tea waste and incorporated Mn. So Mn is the magnetic property. The carbon dot shows that fluorescence. I have incorporated photosensitizer. I have incorporated here photos. Every carbon dot same just our published. I have to show the novel. I have to modify the content. I have to use the carbon dot from somewhere, right? I have shown the 5-6 papers, but I published more than 10 papers on this one. Every time I change the idea, content, but everything carbon dot is same. So here I use the photosensitizer CE6.
The photosensitizer will release the ROS, reactive oxygen species. This reactive oxygen species will help to kill the cancer cells. This is called phototherapy. Because here we use the light, under the light, the carbon dioxide enters with the CE6. The CE6 will release the reactive oxygen sheaves. In the absence of light, they don't release. In the presence of light, they release. That's why we call phototherapy. The phototherapy again two types: photodynamic therapy, photothermal therapy. If the materials of temperature is increasing, we call the photothermal therapy. I am not going to discuss here. This technique we call photodynamic therapy. Light we are using, under the light they are releasing the reactive oxygen species. The ROS will help to kill the cancer cell. Here the total three things are there. One is that MRI imaging because of the magnetic, carbon dioxide are fluorescence. It is showing the fluorescence imaging. The third thing is that treatment, that is the photodynamic therapy. Previously I have explained the targeted drug delivery. That is another kind of treatment. This is the another kind of treatment. So another thing we also use as the antioxidant. Because any disease, antioxidants, free radical. That's why we are having the consume the different kinds of green tea. So any disease, antioxidants are very important because the free radical will damage the cells. So the carbon dioxide we have prepared from the orange peel. So this one I have developed myself. That is a sand bath method. Because when I came back from the India, Taiwan to India, where I started my research, they don't have any research facilities. Okay, then I thought how I continue my research. You people know I left the government job, went for the PhD. When I came back, I don't have the research facilities. That lab have one hot plate. That lab have one hot plate. There is the olden days, even you people use the water bath, right? In the laboratory experiment, the water bath you people use, right? So there is such something, the stand can sustain the temperature. The stand can sustain the temperature. Temperature is the more important than preparing the any material. So then I have prepared a hot plate with the sand. I have given the name sand bath method. I am the first person to give this name in the nanomaterial. So that compound shows the antioxidant and also bioimaging. That work I have continued three works like papaya leaves. This work will show the anti-inflammatory because the inflammation is also one of the major criteria and also anti-diabetic. So this work also published in the good paper. Another one I have prepared from the neem leaf. This work also similar. We have used the antioxidant and the bioimaging. This work it is published in the 2003. I think faculty they know the citation. Within 2 years, it's crossed the 100 citations. This work. It's very difficult to get the citations. This paper within 2 years. So now it is more than 100 citations. It is simply from the neem leaf. I prepare the powder and the carbon dioxide and use. So whatever the materials, all it is came from the nature, crab shell or plant leaves. So all these works are reaching our SDGs goals. Okay. So this is the things. The green synthesis, the leaves or whatever, we can use the green synthesis by using the plant cells, reducing agent, protecting agent to prepare the nanomaterials. Those nanomaterials we can use for the different kinds of application. Directly we can also prepare. I have shown the three kinds of materials, right? Two. One is the carbon dioxide, another one is that calcium. Those different carbon dioxide I have shown in the again the carbon dioxide I have incorporated different elements for a different application. Another one is the calcium. So all these things came from the nature. So this is the conclusion for this talk. If you people have any question on this one, we can discuss 5 to 10 minutes. Later, I will go to the internships and fellowships.
Yeah, because I will go to the other topic, that is the internships. అయిపోయిన తర్వాత అప్పుడు అడుగుతారా ఎక్కడికి వెళ్ళినా స్టూడెంట్స్ ఇదే క్వశ్చన్. నా సార్తో ఎందుకు ఇంటరాక్షన్ పెట్టలేదు అంటారు. ఇక్కడ ఉన్నప్పుడు మాత్రం ఎవరు మాట్లాడరు, బయటికి వెళ్ళిన తర్వాత కావాలంటారు. ఫ్రీగా డిస్కస్ చేయండి, ఎనీథింగ్ యూ పీపుల్ కెన్ ఆస్క్. వెళ్ళిపోదామా నెక్స్ట్? పర్లేదా? ఓకే. We use it as the antibacterial, but we can also use for other application. That doesn't matter. Everything has a chitosan. My goal is that chitosan. That's why, ma'am, if we are going for the biology, that is a different story. I no need to go to the biology. What I need, chitosan. Wastage. The wastage should contain the, yeah. I need chitosan. So the chitosan, why the people have prepared the carbon dioxide from the chitosan. So I have to avoid all the steps directly from the crab. It is there, chitosan I use. What is the crab? That doesn't matter for me. I didn't say here orange pills are anti-cancer drug. Okay. Okay. Yes. తిని పడుకోకుండా ఉంటే చాలా డబ్బులు తగ్గుతాయి. అదే మరి బాడీలో ఎల్లుండి కన్జ్యూమ్ చేయట్లేదు మనం. We mostly focus on the treatments and imaging. If you go to the energy application, so but people are working. See, the carbon dioxide investigated in the 2004, this having the properties replacing the other material. Other materials are showing the very complicated and toxic. Even the carbon dioxide, they are preparing the semiconductor. Yes, yes, yes. So shall we move? I think so. Actually, that Korea, they are providing very good opportunities. That is global intern program. Okay. So this is last year. Now they also open. What is the advantage of this program? You people from the degree students or PG students, you people can go to the Korea 3 to 6 months. You no need to pay 1 rupee. They will pay flight charges, accommodation, and research facilities. If you selected here, there are the many professors they are taking. Okay. There are also other things like short-term internship in Geneva, Switzerland. This is also fully funded. Okay. And ADDA. This is from the German, very popular for the doctorates, masters, PhDs, postdocs. This is another from the GKS, means Global Korea Scholarship. For this, you require the IELTS score around 6 points and small research project. Your faculties can help for this one. So if you secure this one, You can go for the masters or PhDs, they will pay 1 lakh rupees Indian money per month and flight charges and everything. So you don't need to go to any consultancies for this kind of the programs. This is from the Thailand. This program is also they are providing fully funded. Almost all the fellowships you don't need to pay anything. All the things are free, like accommodation, portion fiber, okay, your travel, monthly fellowship, research fund. Okay, even you can save monthly money if you go for the either master or PhD. There are several things like that. This is from the another country, Asian World Fellowship Program. This is from the Taiwan. So in the Taiwan also they have the another internship program that's called TEEP. TEEP. Double T. Double E. Taiwan Education Experience Program. That is also 3 to 6 months you can go similar way. They will provide the everything. If you people want to go to Taiwan or Korea, I can help any good students if there. This is from the Taiwan. Previously, I have mentioned you people require the IELTS. So most of the people, when we are going for the foreign, we think that we require IELTS. But here, no need IELTS. When I go to Taiwan, I didn't have any IELTS course. So we can go without IELTS also, but there is other thing, your project or something you should have to show. Either small paper, something like that. So this is from the Japan. This is from the ISRO. ISRO is also providing the internships. IITs, NITs, all the things, all IITs, NITs, ISRO, ISER, they are providing the short-term internship. You people try to go there and experience the research there. Okay. So now we can see almost 11. All these are fully funded fellowships worldwide. So you people have to find a way how to go. Okay, there are plenty of opportunities without any consultancy fee or you don't need to pay any tuition fee. But we need to find the how to go there. Okay. So this is my research laboratory. Currently we are working on the reproductive system. Okay, male, female reproductive system. But nowadays worldwide the birth rate is decreasing. So we are focusing on that direction. So this is my research group. So my total research team. And these are the fundings like DPT I didn't mention here. DPT, NRF from the Korea and UCSI from Malaysia. There are some other international projects also I am handling. These are some of the my research collaboration. Any research, the collaborations are more important. Okay, when I started back to India, my institute also doesn't have any research facilities. But because of this collaboration, I sustained my research. That's why still I am the leading scientist in the world. Without these collaborators, I am nothing. I can say that. Okay, thank you. So now if people want to ask anything or you can do it. Has interest. Second, we have to see either they have the any IELTS, they are interested to prepare the IELTS or not. Okay, GRE, if you want to go to the US, those things we require GRE, but the students, we don't need to go to the US. So there are some other things. So IELTS is we have the GKS and the Hong Kong, every country have the fellowships. They can apply that. Otherwise, there is options without that one also. And that case they should have some small project or small paper. Then it will the interest, and also we have to find the suitable. Yeah. But we should have try first. Worldwide. నేను ఫుల్ తెలుస్తాను ಇಲ್ಲ అవి ఉంటే మనకి అడ్వాంటేజ్ బట్ లేకపోయినా దేర్ ఇస్ ద వే లాంగ్వేజ్ అంటే మన రీసెర్చ్ ప్రాబ్లం ఉంటుంది కదా, కొంత ప్రాబ్లం ఉంటుంది, బట్ వీ కెన్ మేనేజ్ ఇట్. స్టడీస్ లో అంత ప్రాబ్లం, బట్ లోకల్ గా అవుట్ సైడ్ వెళ్ళినప్పుడు కొంచెం ప్రాబ్లం.
So that we can insert some metals for the increasing the magnetic property of the nanopart, carbon dioxide. What is the technique that you use? A simple lab.
Yeah, we use the hydrothermal and microwave.
In our labs, it is not there. Is there any other technique?
Hydrothermal, it's you need hot air oven. Okay, and 10,000 if you spend, you will get the autoclave, the small one. We generally use the biological big one, right? There is a autoclave small one, which is for 7 to 10,000 in India. So that one you can use it. And you need the temperature 200 to 300 hot air oven. And you require the syringe filter to purify the carbon.
What are the confirmation studies?
I have mentioned, right? Same is not required. Same you need the large surface because of big ones. Same is required to see the small size. And UV, IR, fluorescence.
Changes we have to analyze.
Yeah.
Compare them.
If we are going to the magnetic, we have to see the EPR, magnetic property confirmed. I said is over. If the audience asks the question, I can answer. Otherwise, you people can close it.
Good afternoon to all. I would like to express my heartfelt gratitude for Dr. Gangaraju Gila, grateful professor from South Korea, for spending your most valuable time and gracing the occasion with your inspiring words on sustainable biomedical innovation through nature-inspired nanomedicine. Thank you so much for delivering such as an informative and engaging session about nanotechnology importance, biomedical applications, green synthesis of nanoparticles. Your lecture was highlighting of our program. We are grateful to you, sir. You've given a lot of information about carbon dots synthesis, characterization, and properties. Carbon dots bioimaging, and carbon dots work as a drug delivery, and carbon dioxide as a antioxidants. And also thank you providing internship like this free opportunities, global internship program and global career scholarship. We are very much thankful to you, sir. Your willingness to share your knowledge and valuable session to our students. And finally, on behalf of the organizing committee and all participants, I sincerely thank your esteemed speaker for such as a wonderful and inspiring presentation. Thank you for giving this opportunity. Thank you, one and all.
Person. I request our Vice Principal, sir, Dr. Parangi Ravikumar, sir, and our co-convener also to come on to the dais and felicitate, sir. All the members of the organizing committee are also requested to come on to the dais. request the convener of the program, Dr. Anil Kumar sir, to come and join the program. We'll have a short and quick tea break, 10 minutes. Request all the participants to have tea. Small change. Please stay in your places. Tea will be served in your places. The resource person will be Dr. G. Balakrishnan, sir, Assistant Professor of Chemistry, Telangana University, Nizamabad. I request sir to come on to the dais. Thank you, sir. The chairperson of this session is Dr. G. Mallikarjun, sir, Assistant Professor of Chemistry, GDC Vidyanagar. Thank you, sir. The rapporteur for this session will be Srimati V. Sarita, Lecturer, Department of Zoology. Madam, please come on to the dais. I request Mallikarjun, sir, to continue the proceedings.
Good afternoon to everybody. A warm welcome to Balakrishnan sir. And he is my senior. Of course, he doesn't know me, but I did my PhD in the IICT to Hemant Yadav sir. And just saw your profile, sir. And you did a PhD from Hemant Kamal sir, one of the famous scientists in IICT. And Balkishan sir, now presently working as a board of studies in Telangana University for chemistry department. And he has nearly around 6 international patents for his credit. And he had many publications. Among the famous publications are from Journal of Organic Chemistry, Biomedical, Medicinal Chemistry, ChemCom, EJOC, letters in drug design and decision. Okay. And also, I got a short time, sir. I could not able to read all these things. And sir attended many conferences and participated in many knowledge sharing sessions. And he also handled many projects, many synthetic, organic synthetic medicinal chemistry products. Sir also had postdoctoral experiences around, I think, 3 postdocs, I think, sir. 2 postdoc experiences, sir has, and and also 2 students have been awarded PhD under his guidance, and now presently 3 students are working under his supervision, and many more credits are there for his, sir. It is a very short time, that is I could not able to read all these things. Thank you very much, sir, and thank you, dear participant, for coming here and sharing your knowledge and all these things. Please carry on, sir. Thank you very much.
A very good afternoon to you all. So thank you so much for this opportunity. Just let me begin my lecture on sustainable development goals, the role of life sciences of this conference topic. So I would like to connect my research work and some of the sustainable development goals, how we can interconnect with the medicinal chemistry and these goals. So first of all, let me introduce about this topic. So you know, the world is uh, gaining more and more population and increasing demands, and we have limited resources. How to manage these resources for future generation is very important. Yeah. So in this regard, in 2015, the United Nations members sat together and they agreed to have agenda for sustainable development goals. So they adopted in 2015, and they wanted to reach their goals in 2030. Actually, it's a plan for action for people and also planet and prosperity. It's a very important criteria why these sustainable development goals need to understand. So these uh, goals is built upon 17 number. Those are I am going to show you. And these goals are very, very critical to address global challenges like poverty, hunger, so that they can promote health, education, and environmental sustainability. So these are the 17 global sustainable development goals. The first one is no poverty, zero hunger, good health and well-being, quality of education, gender equality, clean water and sanitization, and this uh, renewable energy. And among these 17 goals, just I would like to interconnect about these goals. This is the third one, good health and well-being. Uh, Mainly, I'm going to focus on this goal and how, being a medicinal chemist, we can fulfill this goal to reach the global sustainable development goals. So first of all, let us try to understand the role of life sciences in reaching these goals. Actually, life sciences can be advancing human health through disease prevention and treatments, and also promotes food security and sustainable agriculture to make sufficient for the population and the generation, future generations. And also, life sciences can enhance the access to clean water and the sanitation. And also, life sciences will be the driving force for consumption and production of the sustainable goal of 12, and understanding and protecting ecosystems and biodiversity. So finally, this life sciences area of life sciences very crucial to provide this knowledge, innovation, and solutions needed to address the global challenges. So mainly, I will show you this picture why this life sciences will be is a plays key role in addressing these goals. Just I am presenting these different kinds of medicinal plants which were used in African continental, and all these medicinal plants will be used to treat the diabetes in their areas. So they may be worked as Ayurvedic or homeopathic and all different kinds of uses they made with this. So let me connect my subject to this life sciences. So medicinal chemistry. So we are going to focus. So medicinal chemistry mainly focus on drug discovery and development, and also it addresses global health concerns and promotes good health and well-being of mankind. And even though we have this very important field of science or life science, so we need to use it properly to meet the needs of mankind. And just this is the uh, compound of salicylic is the stems and roots of salicylic. So where I worked, this is the natural product. We isolated some important medicinal compounds from this, and it can be worked as anti-diabetic agent. And this is about the tree, which looks like Salacia reticulata, which is available in the southern part of India and also in Sri Lanka. So what I want to communicate to you this morning this afternoon, just medicinal chemistry is nothing but we can get lot of medicines from natural sources, especially medicinal plants, and they can be very useful to treat or to address many problems of this mankind. So I would like to focus mainly on good health and well-being of Sustainable Development Goal 3. This is the heart of the medicinal chemistry because the third goal objective is to ensure healthy lives and promote well-being for all ages. Because we know nowadays, so people are suffering from different kinds of problems, and everybody is mainly concerned about their health and well-being, so that the medicinal chemistry is going to address these kinds of challenges. So last time, a Nobel laureate uh, has been interviewed with an interviewer, and he asked a question to that Nobel laureate, how chemistry can be useful to the world. So that's how the question been asked by the interviewer to a Nobel laureate. That lady uh, rightly says that whenever the world is in danger, chemistry comes and rescue. So that was the answer from that Nobel laureate. So recently, in 2019, we have seen the global pandemic coronavirus. So at that moment, so uh, the world is suffered a lot, and many people and scared about those kinds of things and epidemics, pandemics. So at that moment, the chemistry, medicinal chemistry people, the research went well, and they came with solution for that problem. So like that, so medicinal chemistry is the heart of the global health and well-being, we can understand. So it is also a driving force to fulfill this goal in the, even in effective vaccines and diagnostics. In every area, we need medicinal chemistry. So these advancements are critical for reducing child mortality and combating epidemics like HIV/AIDS, malaria, and coronavirus, and etc. And also, they are able to manage non-communicable diseases. So, we need very much advances in medicinal chemistry to design or to improve affordable, effective medicines throughout the world, ensuring health equity to all. So beyond this, medicinal chemistry also impacts in other areas like zero hunger. Okay, this is second goal. In innovation in chemistry supports lot of improved crop protection and also food safety, contributing indirectly to better nutrition and overall health outcomes. The another one is clean water and sanitation. So with medicinal chemistry techniques, we can clean the water and also sanitation so that we can help the world to get out of from water pollutions and pharmaceutical waste and some safe water sources we can provide. And also the other one is responsible consumption and production. The population is growing more and more, so that we need to provide their needs, and we need to develop or we need to improve all the materials to survive them on this earth. Therefore, how to consume the resources and how to product the proper production to the mankind, and also drug discovery. So we know that in drug discovery, we develop new molecules to combat diseases, and also vaccine development, creating immunity against infectious diseases, like coronavirus, and also these diagnostics, advanced diagnostics. So we could able to have so many advanced diagnostics techniques in the field of medicinal chemistry. So this is about, I'm going to talk about my research work, the salacinol, where previously I have shown that slide to you. Salacinol is a potent alpha glucosidase inhibitor, and it is isolated from aqueous extracts of roots and stems of Salacia reticulata. Is the natural product and medicinal plant. So actually, it is traditionally used for the treatment of diabetes in Sri Lanka and also southern part of India. So therefore, wherever I worked in Japan, that group identified this information, and they gathered the material, the stems and roots of salacinol, and they isolated a compound, and they determined the structure, and also they evaluated their biological studies. So the structure of salacinol established by the X-ray crystallographic analysis is quite unique. The structure, it looks very unique. You can see that structure. Yeah. So in salacinol, they have been evaluated its biological activity with modifying its structure, and also they synthesized in the laboratory its structure, and also they have done structure activity relationships by adding some extra substitutions like heterocyclic analogs, and they have reported in very important international journals like Journal of Organic Chemistry and Chemical Communications, like that, because of its activity, prominent activity. Structure oscillate. Why the structure is not? Actually, I have synthesis scheme, but it is not showing. Yeah, this one. No, previous one. Previous one. Previous slide. So, okay, let it be like this. If it is not. Yes. So this is the structure of salacinol. The first one, the number one is, well, this R group is SO3 minus sulfate anion, and if the sulfate anion replaced with hydrogen, it will become neosalacinol, and this is very first and foremost important compound which exhibits anti-diabetic properties after isolation. And this the next one is puncaronol. So the side chain, when it was increased, the name of this compound is puncaronol. When it is sulfate anion, like this, and if you replace this sulfate anion with hydrogen, so that compound will be called neopuncaronal. So these are the very important compounds which exhibits anti-diabetic properties, which are alpha glucosidase inhibitors. They can act against maltase, isomaltase, and the sucrase, those enzymes which are exist in small intestine. And in this structure, this sulfate anion, this sulfonium cation and this sulfate anion makes its structure very interesting and unique. And this sulfate anion and this sulfonium cation uh, partially forms some ring at this place, and which can be confirmed by analytical techniques. So it was discovered that it was strong alpha glucosidase inhibitor activity, and when compared to voglibose and acarbose, which are already in clinical use, and the total synthesis of the salacinol was accomplished by two groups by the use of nucleophilic ring opening reaction, and one is our Osamu Muraoka group in Japan. So after synthesis, the intensive studies on structure activity relationships has been uh, studied, and they found the more potent compounds in these structures. The synthesis. So I want to show you the synthesis of this compound. Yeah. So first of all, this arabinose was protected with the acyl group, and this protected compound will be protected. This primary hydroxyl group will be protected with the PMB, paramethoxy benzyl chloride. The first step, in the presence of these reagents at 60 degrees centigrade, it produces about 92 to 95 percentage of yield of this compound. The second step, to this compound, we can attach these substituted aromatic rings at this secondary hydroxyl group in the presence of these reagents. Okay. In the case of nitro, the reagents are very different. So in the case of different substitutions, we have used only sodium hydride, but in the case of nitrate, so this sodium hydroxide will be the successful to produce the uh, analog. And after that, epoxidation of this compound, removal of this acyl group and epoxidation, and this epoxidized product will be coupled to this sulfonium compound and produces this compound. This the compound, and after deprotection of PMB will produce this final target compounds with the different substitutions like ortho substitutions, meta substitutions, and para substitutions like this CS3, ortho CS3, ortho chloro, ortho CF3, and ortho nitro group, and meta CS3 chloro, CF3, and nitro groups. And these are the compounds and analogs we have prepared, about 12 to 15 analogs we have prepared like this. So after the synthesis of those analogs, we have evaluated their biological activities. IC50 values has been shown like this. Okay. So in these compounds, you can observe that this 7K compound, orthonitro compound, exhibits more potency compared to the other compounds. Okay. This 7K compound. These values are very highlighted compared to this already standard drugs is voglibose and acarbose. If you observe this 1.2, but in the case of our synthesized compound exhibited 0.13. So concentration, it is uh, showing very good effect. And also chloro substitution is ortho CS3 and this orthochloro and H7H, 7B, 7E, and 7H and 7K. So these are the four analogs are synthesized compounds exhibits very good or very promising activity compared to voglibose and acarbose. You can observe these values. So these compounds create very interest to develop further. So biological evaluation informs us that all the compounds showed superior activity to the original sulfate, sulfonium sulfate. And among the sulfonium salts designed, this 3 PO ortho nitrobenzyl moiety was found to the most potent. That was our research discovery, and it is about 40 times as potent as compound 1 sulfate ion. So that is the thing we have discovered with that. Why they are not? Yeah. So these are the silico design studies. Before we synthesize, we done these studies. In these compounds, different compounds, 7K, 7B, 7C, like these compounds, how these uh, synthesized compounds interact with the these amino acid residues. So here, the gray dotted lines will show the hydrogen bonding, and the red dotted line shows the this anionic bonding, and the green dotted bonds will show the hydrophobic interactions with the amino acids of these compounds. So among them, this 7K C will be the most effective, will be supported by these studies. So research outcome, in our research outcome, so we used this in silico method, and we found that more potent alpha glucosidase inhibitors 7B to 7M, then the seed compound salicylic. The seed compound salicylic will be isolated from the natural product Salacia reticulata. Okay, so these compound directly used by the people, tribal people in the forest for diabetes disease as the treatment for diabetes. So this works effectively. So what we do, our research group, what we do, so we took that salacinol uh, stems and roots of that salacinol, and we isolated the salacinol compound, and we developed that compound into different analogs like poncarinol, neoponcarinol, protanol, like that. So those compounds exhibit so more potency than this one, salacinol. So that's what we developed in our research work. So finally, the sulfonium salt of 7K was found most potent among other cyclic sulfonium family, a new class of glycosidase inhibitors synthesized so far. Challenges. So what are the challenges we have in, had to manage these goals. So this broader adoption, encouraging wider implementation of sustainable practices in medicinal chemistry and research globally. And another one is, so navigating the complex interplay between innovation and cost effectiveness and environmental responsibility in drug development, developing eco-friendly and scalable solutions to meet the growing global demands for medicines. So we need to create a sustainable and healthy world with the help of medicinal chemistry, future medicinal chemistry. For that, we need to apply some different strategies so that we can align with the sustainable goals. And the key to this vision is the expansion of green chemistry. You know about green chemistry education, and also we need to fostering collaborations with academic and industry people and government sectors. So ultimately, a world where medicinal chemistry not only cures diseases but also actively protects our planet and nurtures vibrant communities. Conclusion: medicinal chemistry is essential for achieving good health and well-being and environmental and economic sustainable development goals by 2030. Okay, it involves in drug design and manufacturing, already transforming global health and industry practices. So finally, I would like to show about my research experiences. So I had a privilege to meet EJ Kore, the Nobel laureate from USA. Okay, and we have met in international conference in Japan, Tokyo. And this is about our research group in Japan. So when I received Young Scientist Award, Early Career Research Award, so I was being awarded, appreciated by the Vice Chancellor and Registrar of the university. Thank you so much. Yeah.
Thank you very much, sir. Students, any questions? Any clarifications? One of the prominent subject in chemistry is medicinal chemistry. We know our life expectancy has grown up from around 40 years to now around 70, 80 years has grown up nearly due to the advancement of medicinal chemistry and latest technologies involved in the drug discovery in medicine of drugs because nowadays the technology's involvement in the drug discovery has enabled us to discover new drugs in a very short span of times. We know already the drug discovery involves around 15 years of time to get the market. During the COVID time, we know already IRCT had done a good work so that people can benefit through that one. The same manner, sir also did a very fantastic work on the anti-diabetic drug. So thank you very much, sir, for your work and explanation. And I think sir has also been awarded as a Young Career Award also, and he has many 4 awards have been there for him, and he has industrial experiences, postdoc experience. I think it is more than sufficient to handle any type of laboratory work from microgram scale to kilogram scale. I think you covered around microgram scale to kilogram scales also, sir. Thank you very much, sir, for your beautiful delivery lecture. Thank you very much.
respected dignitaries, esteemed guests, speakers, participants, and dear colleagues. A very good afternoon to all. It is my honor and privilege as the rapporteur of this session. First and foremost, I would like to extend our heartfelt thanks our chief guest, Dr. Balakishan Sir, Assistant Professor of Chemistry, Telangana University, Dichpalli, Nizamabad, for gracing us with your presence and delivering an inspiring address that set the tone of for the seminar, sir. Yes, sir, your speech was really thought-provoking and truly inspirational, sir, by explaining the impact on sustainable development goals 2, 3, and 6, and 12. The chairperson of our sincere thanks to go to our chairperson of this session, Dr. G. Balakrishan Mallikarjun, sir, Assistant Professor of Chemistry, TDC, Vidyanagar, for valuable words and encouragement, sir. Thanks for the giving this opportunity. Thanks.
Thank you, Dr. Mallikarjun and Dr. Balakrishnan. Now I request our team, Jyotsna Madam and Shivrani and their team, to come and felicitate the guest later. Yes. Together, okay, fine. Okay. Now I thank you very much for this session. So next we will be going to the next session. The next session will be on the intellectual property rights. Dr. Y.V.R. Prasanna Kumar would be the chairperson for this session. Meanwhile, there are few announcements. After this session, all the lecturers will go for lunch. Immediately after the lunch, again you have to come back here. We'll have valedictory session. Okay, with the valedictory session, we'll be concluding the today's national seminar. Okay, all the lecturers, remember you have to go to the library. There lunch is provided for you. Okay, afternoon only valedictory session will be there. And one more announcement is from IPR. The lecturers who have attended this session will be provided certificate that you have participated in the IPR lecture. This will be useful for your career advancement. Okay, is this useful for the students too? So they have to register. Okay, fine. Maheshwari Madam and Sarita Madam, please note down the names or send a notebook to the students. They will write their name, class, and their roll number. So that they will be given certificate for this IPR. Okay, and the lecturer, somebody, maybe Sarita Madam can take care of the lecturers. Those who have attended only will be receiving the certificates. That's very clear. So without attendance, we cannot give any certificate. We have come to the last technical session. I hope till now, definitely there is a benefit to the students and the teachers and the research scholars and those who are presented online also. So for the last session, I would like to invite Dr. Y.V. Prasanna Kumar. Sorry for the delay. I request Dr. Y.V. Prasanna Kumar sir to chair this session. I invite Dr. Ayodhya onto the dais, please. And I also request Dr. Krishna sir, lecturer in botany, onto the dais as a rapporteur. I request Dr. Y.V. Prasanna Kumar to continue the proceedings.
Good afternoon. At the outset, I would like to express my apologies for being late and keeping you on waiting. It's my pleasure to introduce Dr. Ayodhya Dasari, who is presenting a seminar on intellectual property rights. And I would like to introduce him. He had been to this college 2 years ago, and he took the same seminar here for all the BCom students and BCom and BBA faculty. And now we thought of having it for all the faculty here so that we may understand what intellectual properties are. Okay. So Dr. Ayodhya Dasari is working as an assistant controller of patents and designs. He is from Nalgonda. Actually, he is son of the soil. And మీరు రోజు చూస్తూ ఉంటారు కదా, ఏంటి అంతటి శ్రీనివాస్ సారు పోస్ట్ చేస్తూ ఉంటారు పేటెంట్స్. ఆ మధ్యలో నేను కూడా ఒకటి రెండు పోస్ట్ చేశాను. అవి ఏంటి అవి అసలు? ఆ పేటెంట్స్ ఇష్యూ చేసే ఆఫీస్ ఎక్కడ ఉంది? హూ విల్ ఇష్యూ దెమ్ అండ్ హూ విల్ డూ దట్ వర్క్ అంటే అయోధ్య దాసరి గారు పనిచేసే ఆఫీస్ చేస్తారు. ఇట్ ఈస్ ఇన్ చెన్నై. So if you want to have patents, you have to approach sir's office. and he is the controller of those things. అన్ని సబ్మిట్ చేసినవన్నీ కూడా పేటెంట్స్ కావు కాబట్టి you should get some knowledge from him how to get patents. So he has a lot of experience as an examiner of patents. Currently he is working as a NEPAM officer. NEPAM means National Intellectual Property, okay, awareness mission. Okay. So he finished his MSc and PhD in chemistry from Usmania University, Hyderabad. He was best research student during his PhD and published 95 research articles. 95 research articles and presented papers in more than 45 conferences like this. He has 6 patents, not patients. He has 6 patents on his name and published. 2 books are also published by him in the field of chemistry, especially material chemistry and engineering chemistry. Now he is also working as a reviewer for the research articles for review and for more than 68 research journals, including American Chemical Royal Society of Chemistry. Now has been working as an expert in training intellectual property rights law from renowned institutions like Wipro, EPO, USPTO, and other organizations. Okay, and it's a great pleasure to have Sir among us to present a seminar on intellectual property rights. So thank you so much. This is your time, sir. I hope you will enjoy the session. Thank you.
Good afternoon, everyone. So due to lack of time, I will finish within a very short time. Myself, Dr. Dasar Ayodhya, Assistant Controller of Patent and Design, Patent Office, Chennai. So majorly my office doing the giving the patent rights for different innovations. It's not like a specific category. It's a very broad category. It's useful for the physical sciences or chemical sciences, the life sciences, whatever it is. So different of intellectual property rights are there. So simply I will give the brief introduction and what is the benefits of this I will explain. If you need more information, please contact if any time. So these are the content I would explain in this program or seminar. That is the what is the intellectual property right, what is the importance and significance and examples. And second one is the very young innovators, means 3rd class, 4th class, 5th class students also got the patents in our India. So I would show some pictures. And third one is the what is the procedure to file a patent in India in different of innovations. The third one. And fourth one is the what is the intellectual property rights role in life sciences or chemical sciences. And the last one is the it is also some career progression also is there for the student and faculty. So these are the content I would explain in this program seminar. Yeah, the first one, if anybody wants to file a patent, don't publish in any journals. If you publish your journal, any your invention, patent office cannot accept the inventions. Before going to publish, you can protect our intellectual property rights. So it can be accepted only before the awareness of it, before the publishing journals. After publishing journals, it cannot be accepted as a patent. So these are the differences between the research paper versus patent. Patent is issued by the government of India, like a patent office. So it will give a techno-legal document and it will give the patent rights to only the invention and inventor. But research paper is not like that. The copyright goes to journal only, but not of you. That's why don't go publish before the getting the patent rights of your inventions. So what is the meaning of intellectual property right? Simply we know the properties like a land properties or house properties. Those are the conventional properties. But intellectual property rights means only can be intellected by the human mind. Only human mind can create the intellectual property rights for your inventions. So these are the examples. So what type of inventions should file intellectual property right? For example, students and faculty can seek only technology development, only new drugs can be given the patents or IP rights. It's completely wrong. You have a eligibility to write any new project or new poem or new lyrics or new stories, new drawing, new photography or new logos, new designs, new mobile apps. Anything should file intellectual property right in different categories. Patents only technology and process or products. But these are the different are there. Different of intellectual property rights are there. Not only patent is one of the IP only, not entire IP. Who can create intellectual property rights? Everybody should create, like a student, a faculty member, or college, or university, or company, or laboratory, or any organization should file intellectual property rights. Then, what is the benefits? What you get the benefits of intellectual property rights? These are the benefits. You can get the recognition, or you can earn the money from the license to the companies, or agreements to technology transfers, or new innovations, and increase the value of money as well as country and global value also will be increases. So these are the benefits when you get the intellectual property rights. So simply give the example of importance. This picture shows simple bike accident on the road, right? So what is the treatment? Doctor will tie around the neck with hand. It's a simple treatment, but a fifth class student can think differently. Why such type of incident got this such treatment? He think differently, and he got the different product has been invented. He got the patent. So this boy name is Adi Kumar, fifth class student from Tamil Nadu. He has been developed a new product. The product is wristband with supporting of hand. Such type of accident is one of the treatments. There is no neck tag, there is no hand tag. So that product belongs to that boy only. He got the patent for developing such products. Anybody wants to manufacture such product, those get the permission from the boy. Without the permission of the boy, they cannot be manufactured, they cannot be selling or any marketing. And one more thing, we know the Facebook. So, at the last 2-3 years back, Facebook company received a logo, that is a Meta logo. For reason, Meta logo only registered intellectual property rights. That's why Facebook paid 440 crore rupees to Meta company just to logo. Meta company has registered the logo as IP. If in case the Meta company does not take the IP and the logo, everybody can use. Specifically, Meta company registered that logo and intellectual property right. That's why Facebook company paid 440 crore rupees to the Meta company to receive the logo. That is the importance of intellectual property rights. So these are the different types of intellectual property rights. Here, several types are there. Among them, patent is just only one thing. Here, patent, design, trademark, copyright, geographical indication, semiconductor integrated circuit layout, and trade secret. These are the 7 different of intellectual property rights. So I will explain one by one in detail. The patent. The patent one is a completely technology development or new product or new process only gives patent. Okay. So I will give some examples. Yeah. These pictures are some eligible to get the patent filing. Those pictures are very rare. So this first picture is look like umbrella, but it's not umbrella, right? So that product is eligible to file a patent. So the second one is the digital screen, and third one is the drones technology, and fourth one is the marketing, digital marketing, and fifth one is the cap with light. We only cap, yes, but cap with light is new invention. Such product is eligible to file a patent. And the new kids and new cricket field is how can a batsman take it out means nowadays this is spark of the light. That's also one of the technology. So these technologies are eligible to file a patent. Second one is the design. Design means any new design like a floor designs or dress materials or new design of the pen or new design of the toothbrush or bags or wallets, whatever it is. That's a new design is eligible to file a intellectual property right under design category. So these are some examples. New toys, new water bottles, new wall designs like this. New shape of water bottles, new shape of shampoo bottles like this. Any existing material with new design is eligible to intellectual property. Nowadays, it's a trending marketing also. Marketing is a digital, means a design marketing. So new design of the dress materials, new design of the slippers or sandals, new design of the wallet, toothbrush. So everything will be new of the existing one is eligible to file intellectual property right under design category. Third one is most appropriate for the commerce student. So simply trademark is given for the logos, brand names, symbols, and slogans. Simply how you are identify a person means based on their logos. Simply you can go in a market. You have to purchase a shoe. It's a based on Nike or Adidas or Puma like this, right? Based on logo. That logo will use the trademark. Recently, in market, some of the fake products are coming. So Puma is Puma means the wording is different. Adidas is Adibas. It's a different of the logos and designs. Those are the fake products. So if you have any eligibility to create logo, you can file trademark. So simply nowadays, everything the fake product also coming. Distillery water bottle, it's coming Distillery. So different of fake products are coming. So these such avoiding of fake products should take intellectual property and trademarks. How you can recognize the product is trademark or not? Simply you can go any market. Just you can take one product. It has been mentioned TM or R around the circle. That product is trademark products. Remaining are fake products. Copyright. Already education institutions very well known about this copyrights. So copyright we give only literary works, musical works, artistic works, and cinematography and sound recording, photography. Such things are eligible to file copyright. Simply, that last 2 years back one movie came, Something Right. Yes, what is the motto of this movie? Copy of a novel. Right? Hero, a novel has been copied. He has been imprinted as a criminal case. Why? Because that novel has been copyrighted by some other. But you don't know. Simply he copied from that novel. Automatically he got the criminal case. That's why if you have any ability to write a poem or whatever book or whatever lyricist or whatever cinematographic sound recording, you are able to file copyrights. Once you get the copyright, nobody can copy without your permission. Simply you can write a poem or you can write a book, simply go to the sale of the market, anybody should copy. Before copywriting should be eligible to take only copy. After that, you have to get the copyright permission. Nobody will copy. So how you can identify that product is copyright or not? Simply see around the circle. Simply after this program, you went to your library. You can take one book. And a lot of the last page, you see around the circle means that book is copyrighted book. Nobody can copy. If it is not there, you can copy. That's the copyright of this musical and educational thing. The fifth one, geographical indication. I am saying this 7 type of intellectual property right. This one very specific. Why? Because single person cannot file GI. A group of members or any organization only should file. Why? Because Tirupati Laddu available anywhere in India, only available in TTD. Why? Because TTD organization should file GI. Like Gopili Veena, like Kondapalli Toys, Warangal Dairies, and Darjeeling Tea, and Venkaragiri Sari, Adilabad Dhokra, Hyderabad Halim. This is not a specific person. One organization or geography of the place only should file GI, geographical indication. And some more examples also there. Guntur Mirchi, Basmati rice, Darjeeling tea, Kanchipuram silk saree, Alpinos mango, Nagpur orange, Kotapuri chappal, Digni Bhujia, Agra peta, Rasagulla, Rajasthan marbles, Sona halwa. Those are all GI products based on eligibility of the geography based on the organization. We have Pochampalli saree. Pochampalli saree also previous So different nowadays, based on the origin, you should file GI, but not a single person. Organization should file. And the sixth one is the semiconductor integrated circuit. In our India, is a very bad condition in this position. There is no any semiconductors developed across the global wide. Only Taiwan has the first place in this position. Means you have to eligibility develop any new designs, layouts only eligible for this category. And the last one, trade secret. Trade secret means any recipe like Coca-Cola, Sprite, Thumbs Up have same taste. No different taste, right? Based on ingredients. But the company cannot be issue the ingredients, what is the content is there. That's the trade secret. Why? Because everything should know, they have to prepare, they have to manufacture. So that's why their marketing will be down. That's why trade secret also should one of the IP also there. For example, your mom, your daughter, whatever it is, should prepare any one of the recipe in a kitchen. That's a new one. You can take this trade secret also. So I was going simply one product, IP everywhere means a lot of IP are there. Yeah. In a laptop, in simply all IP should be covered. Why? Because symbol is a trademark. Working mechanism, patent should file. Body of laptop, design. And Apple or Oscar or XP's logo is there. That logo is trademark. Manual, copyright. Integrated circuits, that's semiconductor integrated. Means a single product, multiple intellectual properties are there. So the best example is laptop. For example, you have a new invention, but you don't know how to file. These are my office structures. Government of India, only 4 offices are there in India, like a 4 directions: east, west, south, north. So here, Delhi, Chennai, Mumbai, Kolkata, 4 directions, 4 offices are there. In the Nagpur is the training for the intellectual property officers. These are the total Indian IP structure. Second one is very interesting for the students, inspiring young innovators. The first picture, in general, chessboard at a time, how many members can play? 2 members. But this boy, Jaipur, means age is 9 years. He has been developed a new chessboard. At a time, 6 members can play. Do you know about this? He got the patent for this development of board. Any sports company should manufacture such type of board. They will get the permission from the boy. Why? Because he has the technical mind to manufacture such board. His age is 9 years only. The second picture, Sharat Sumesh. His age is 4 years. He has been invented new robot. He got the patent for this development technology. His age is just 4 years only. And nowadays, UKG. The top one, Gursimran Singh. He has been intermediate second year. He has been developed ice cream. He got the patent for the product. Anand Dadar, he has been invented goggles for the blind people. He got the patent for such technology. His age is class 11. It's under 16 years of age. Ramya Josh, she has been invented in Kerala washing machine without electrical energy. How it is possible? That machine is complete with the cycling. She do cycling, automatically machine is operating. Here 2 benefits. There is no electrical energy consumption. There is no separate gym. She has itself has also some physical maintenance. She has been such developed a washing machine. He got the patent. She got the patent. The last one, Tenit Adityan. He has been developed banana leaf technology. In nowadays, when we go to so many functions, what happens? Plastic plates, plastic glasses, plastic water bottles, everything. So those are not biodegradable. But this Adityan has been developed banana leaf technology, totally banana leaf only. Plates, glasses, gift boxes, everything. After use, it's easily biodegradable. There is no pollution. For this development, he got the patent for this banana leaf technology. And she is MC Vaishali. Age is 3 years only. She got the patent. It is the age of 3 years only. Nowadays, maybe Anganwadi school, I think, or LKG or nursery, I think. But she got the patent in India. Very youngest patent holder in India, MC Vaishali. age is 3 years, she developed a rescue flooded house. In coastal areas, floods will come. Automatically, the water floods into the house. So she developed a new house construction without entering into the flood water into the house. Based on this invention, she got the patent. And mid-cool refrigerator. In our refrigerator in our homes, continuing with electrical energy, right? At this mid-cool refrigerator, there is no electrical energy. Simply mitti means matti and clay materials with wood. Those materials are helpful to manufacture refrigerator. Without spoil, 72 hours, your food is very safe. There is no electrical energy. Simply matti and wood only. This is also one of the new inventions. This got the patent. And not only ancient days, in previous days also, Peter Chawlis, wine surfing, he got the patent at 12 years old. Louis Braille reading is age of 15 years. And George Nelson and trampoline at 16 years age. Alexander Graham Bell found a telephone at 18 years old. And George Westhoff and rotary system engine at 19 years old. And Vincent Pascal, mechanical calculator found at 19 years old. I think those are all under your age only, right? So, dear student, if you have any thoughts in your invention to develop such products, you got the patents for such invention. You have to think in this mind in the way. Okay. The third one is the patent application procedure in India. So, simply I will explain this. Only the two processes are there. Online and offline is there. If you want online, simply you can log in with your username password, you can find. Okay. Otherwise, offline means your document entirely you have to take and signatures should post to the patent office. You can consider the post also and give the process of your patent application. So different applications are there. So among of these, who are the first filing? Those are eligibility. For example, two processes are there, X and Y. Those two are the same invention. Who approach the patent office first? That's the value. After one day, also the Y person came to the patent office, file the patent, it's invalid. That's the benefit of the priority date. Priority date means peer date. Who is approaching first, then those are the eligible to file the patent of India. So I will show some images quickly. So different patent applications are organizing in India. So if you want to file a patent, like your chemistry or physics record, what do you write? What is the aim? What is the apparatus required? And what is the procedure? What is the result discussion like this? So same patent application also like first title, any reference are there, any background, summary, description, and any drawing, line, abstract. The step by step you file, and your patent office will be examine your application in the patent grant or so your inventor and applicant. So if you want to file a patent, some forms are required. So due to lack of time, so if any want to file patent, please approach me. I will give the full guidance. This is my government India patent office website. If you want to know more information, just you can go IP India website. IP India, Intellectual Property India. You can get the more information about all type of intellectual property rights. So these are the patent sensitive databases. If anybody interested, any one of the link you should click, you can get more information about the patents. So after filing a patent, these are specifications. After publishing, after the grant. These are the two differences. This is the publishing certificate, this is the grant certificate. Once you grant the patent, 20 years of lifetime. After 20 years, it will be generic. Everybody should use without your permission. So IP role in chemical and life sciences. So lots of life sciences patents also filed. Those are give some examples here. Different type of patents have been granted in different life sciences. So alcohol, drug addiction formulations also grant a patent, and any drug delivery is also patent granted. Different of life sciences products and chemical as well as physical also got the patents in India. So those information entirely is available in IP website. So the finally theory development. Any student or faculty so interest in this IPR regime Lot of benefits are there. Once we, in India, so many universities are offering IPR courses, intellectual property act courses. Dear students, please listen. If you have totally our mindset is PhD, MSc, PhD, government job like this. So please aware this. This is a very bright opportunity for me who are interested in IPR. Once you did IPR course, lot of opportunities are there. Like you become a patent agent, you become a patent drafting specialist, you become a patent analyst, you become a any law graduates in different of IPR. Otherwise, you can become a government service like me, IPR government service job, and IPR policy, IPR cell or any institution. Lot of opportunities are there. If anybody, any students interested in this field, please contact me. I will give the full guidance to you. Okay. These are some useful links. If you want to know more information, already in your college website is there. Those I mentioned in the total PPT. Anybody wants to take an approach it. Yeah. This is the final conclusion. If you have any question, please ask me without hesitation.
So please give a hand. In a few minutes, I explained a lot. And thank you very much, sir, for a wonderful session here. We had learned so many things. And before going for any questions, can you see this here? If you file a patent, you get a certificate like this. I'm proud to have such 2 certificates. Do you know what is my subject? English, not science or something else. An Englishman has 2 patent certificates like that. And one of our chemistry sirs, associate professor, Mr. Antak Srinivas, has more than, I don't know number, but he has more than 10, as I know, from our college. We have been posting them in the groups, but we are receiving poor response. I hope in the days to come, we will also be presented such certificates with the help of our dear presenter, Mr. Dr. Ayodhya Dasari. Once again, can you give a big round of applause for him for his wonderful presentation? And if he explained so many things, one of the things I just want to tell you the effect of it. In one of the topics was on trade secret. Trade secret గురించి చెప్పారు. 2006 ఒక ఆమె జోయా అనే ఆమె ఏం చేసిందంటే షీ వర్క్ ఇన్ కోకోకోలా కంపెనీ. ఆమెతో పాటు ఇంకా 3 కలిసి ఆ కోకోకోలా కంపెనీ ట్రేడ్ సీక్రెట్ అంటే ఆ కోకోకోలా డ్రింక్ ఎలా ఏం మిక్స్ చేస్తారు ఏంటనేది ఉంటది కదా రెసిపీ, ఆ దాన్ని దొంగలించి పెప్సీ కంపెనీ వాళ్ళతో కాంటాక్ట్ అయ్యి మేము కోకోకోలా కంపెనీ ట్రేడ్ సీక్రెట్ మీకు అమ్ముతాము, $15 million కి రేట్ కుదుర్చుకున్నారు. పెప్సీ కూడా ఏం చేసింది, సరే అని చెప్పి చివరికి ఏం చేసిందంటే FBI ఫెడరల్ బ్యూరో ఆఫ్ ఇన్వెస్టిగేషన్ మన సిబిఐ లాగా FBIకి ఇన్ఫర్మేషన్ పంపించింది. కోకోకోలా వాళ్ళు కూడా పెప్సీ కంపెనీ వాళ్ళు చెప్పారు. సో FBI ఏజెంట్స్ ఏం చేశారంటే వాళ్ళు పెప్సీ కంపెనీ వాళ్ళలాగా వచ్చి వీళ్ళతో కూర్చొని స్టింగ్ ఆపరేషన్ చేసి వాళ్ళని పట్టుకున్నారు. సో ట్రేడ్ సీక్రెట్ అనేది చాలా 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 పవర్ఫుల్. సో మీ దగ్గర ఏమైనా ట్రేడ్ సీక్రెట్ మామిడికాయ పచ్చడికో ఏదైనా సరే, ఇఫ్ యూ హ్యావ్ సమ్ రెసిపీ అండ్ ఇట్స్ a trade secret, you can file and you can get that. మీకు తెలుసా పసుపు పేటెంట్ ఎవరి దగ్గర ఉందో? మన దగ్గర లేదు. ఇట్స్ విత్ అమెరికా. Because we failed to file it. It's our product and we failed to file it. Now that patent of haldi is with USA, not with India. So with this brief remarks, I once again thank you, sir. If you have any questions, you can ask. You are most welcome to ask. Yeah.
Sir, how do we check our concept already exists or not? Where we should check whether our concept is exist or not, whether it is new or not.
in IP India website, INPAS is there. INPASS. INPAS website is there. In INPAS, already existing patent details, everything is there. Not only India, global wide is there. So you have to search what you want to search. Just simply keywords you enter into this. So automatically you will get the total information about this. So if it is there, then you can modification of your invention. If it is not there, as it is, you should file the patent. Yeah, any more questions, please?
Yes. Thank you, sir. You have explained about manipulators of logo and selling some goods in the market. So what will be the intensity of punishment to that kind of situations?
Yeah, very nice question, sir. Actually, the recently, like some Fevikwik company, like Fevikwik is a recently fake product is Fevikwik is coming. Fevikwik is a Fevikwik, but just simple of number or letters will be different only. After that, Fevikwik company has been found it's a fake product. He has been infringement case. Infringement case means automatically it's an existing product. So after infringement case, the court has been given the orders. Total for Fevikwik company goes to Fevikwik. What they got the benefits, what their market entirely shift to Fevikwik.
So total Fevikwik company is handed over to the Fevikwik company.
Without single NP, they are not having this Fevikwik. Total Fevikwik company handover by the Fevikwik. Why? Because Fevikwik is already patent product, the IP. So that's why court also given the orders. After 20 years, it will be generic. Everybody should utilize. If in case that Fevikwik company is more interested to in the market, they have been renewal it up to 60 years.
Thank you, sir.
Yeah, please, any more questions from the student side? Okay, can I conclude? Thank you for giving the opportunity for this organization. Thank you so much.
Okay, thank you, sir, once again for all the explanations and for the clarifying our doubts also. Now I request Sri Krishna, sir, to present proposed vote of thanks.
A very good afternoon to one and all present here. I would like to extend heartfelt gratitude to our resource person, esteemed Dr. Dasari Ayodhya, Assistant Controller of Patents and Designs, Department for Promotion of Industry and Internal Trade, Ministry of Commerce and Industry, Government of India. You are given wonderful information about intellectual property rights. After listening to the address of Dr. Ayodhya sir, our entire team gained more knowledge about intellectual property like patents, design, trademark, copyright, geographical indication, trade secret, semiconductor, integrated circuits, layout design. We are very much thankful to sir. You are appreciate your willingness to share your knowledge and valuable session on IP everywhere, inspiring young innovators, patent application procedure in India, IP role in chemical and life sciences, carried development, sir. Sir, indeed your words, your voice, your lecture and your expressions have inspired my students and also me. I would like to thank to our principal sir, Dr. S. Upender sir, this national seminar convener, Dr. Anil sir, and all the organizers to given this wonderful opportunity to me. Thank you, sir. Thank you so much, all of you. Thank you.
Thank you very much, and I hope you enjoyed this, and we'll proceed for lunch. No? Oh, sorry. Over to convener.
Now it is the time for felicitation of the guests. I request Dr. Balakrishnan sir to come onto the dais, and I request my organizing team to come forward with the shawl and bouquet. Thank you, my dear team. Now it's time for lunch. I request all the participants to move for lunch. Immediately after the lunch, come back here by 2:30. We'll be having valedictory session. Okay, sharp 2:30. I request all the participants to come back and the lecturers, all the lecturers to come back here immediately after the lunch. Thank you very much. It's a great pleasure that the national seminar has been held in our college for 2 days. To achieving the desired objectives of the seminar, everyone has done the research on the various topics and submitted the research papers and worked hard to make the national seminar in a successful manner. Since the beginning of this national seminar yesterday, various sessions have been held. Finally, the closing ceremony is to be held. Therefore, on behalf of the organizing committee, I would like to express my gratitude to the guests who attended this closing ceremony. In this closing ceremony, now I request Dr. S. Upendra Garu, Principal, Nagarjuna Government Degree College, Nalgonda. For this session, for this closing ceremony, now I am request today's chief guest of this closing ceremony, Dr. Nagajyoti Garu, Pro Vice Chancellor, West University, Andhra Pradesh. I request you to come onto the dais. Thank you, ma'am. I request Shirisha Madam, Assistant Professor of Botany, please present a badge to today's chief guest. Now I request Madam Shivarani Garu, please present a badge to our principal, sir. Now I am inviting today National Seminar Convener, Dr. Anil Kumar M. Please come on to the dais. Today guest of this valedictory ceremony, Dr. YVR Prasanna Kumar Garu, Coordinator IQAC, NJ College, please come on to the dais. And another guest, Dr. Parangi Ravi Kumar Garu, Vice Principal, Nagarjuna Government College, please come on to the dais, sir. And now I am inviting Dr. Antati Srinivasan Garu, Vice Principal, Nagarjuna Government College, NG College, Nalgonda, please come on to the dais. And today's National Seminar Co-Convener, Anil Kumar Bojja Garu, Doctor, please come on to the dais. Organizing Secretary of today's National Seminar, Dr. B. Nagaraj Garu, Control of Examination, NG College. Now I am inviting for this valedictory ceremony, Head Department of Zoology, Dr. Josna Madam, please come onto the dais. Now I am requesting Head Department of Botany, Madam Shivarani Garu, please come onto the dais. Thank you, ma'am. For this valedictory ceremony, now I am requesting Dr. Anil Kumar M. Garu, Convener, Today National Seminar, please announce the report for today's seminar. Thank you, sir. Good afternoon, all dignitaries on the dais and my colleagues and the participants from various colleges, students, scholars, teachers, and my dear colleagues. A very good afternoon. I want to thank the Council for Higher Education and RUSA, PM RUSA, and Telangana College of Sciences for granting the grant to conduct this national seminar and for all these things. So definitely I must receive good support from the principal, which I have received, so that we could get some funding so that we can conduct this program. So I want to thank the principal sir and along with all these funding agencies. First, let me start the report with the first session, that is inaugural session. Distinguished speakers and senior most professors from the University of Hyderabad, Professor T. Radhakrishnan, who is the pioneer of Asia in nanotechnology. Way back in 1990s, after 2004, the work began all over the world in the nanotechnology. From then, Dr. T. Radhakrishnan was very active, and he, from the scratch, he gave a lecture so that every participant could understand the importance of nanotechnology and its applications in day-to-day life. And followed by the alumnus of this college, Dr. Ramachari sir, he has spoken about sustainable development in organic chemistry. The chemistry lab is very hard to see and hard to go into the lab for non-chemistry students because of the smell or whatever reactions that we conduct. Many people may not like, but this has to be changed. It should be a sustainable. That was the focus of Dr. Ramachari sir, and he has spoken about the enzyme catalysis, which is actually the sustainable development goals. Actually, it is reached by the human body. That was the green chemistry. That green chemistry he was trying to see in his laboratory, so that the world may go into the sustainable development. And followed by the third session, Dr. Shamlal from University of Hyderabad. Shamlal is a biotechnologist. He was again into the animal biology applications of nanomaterials. So the whole theme of this seminar, the sustainable development goals, and how a life science student or a scholar or a teacher or a professor or a scientist, how we can achieve these goals. So these all are touched by Dr. Shamlal. Then in today's session, Dr. Gangaraju, who is a brain pool professor, is working in South Korea, Anchung University. He has stressed upon the importance of green chemistry, and he is from biotechnology background, a biologist, application of nanomaterials. Once, see, when I say it's nanotechnology, it doesn't just belongs to physics or chemistry, but it's a natural science. That's what he was focusing upon. The nature, it has already technology, but now science only, it can observe and explain the things. There are many hidden things in the nature, but we are trying to develop it so that we can understand the nature. That's what he has focused upon. And he also tried to impress on the good health sustainable goal, good health goal by his research work. And later, a professor from Telangana University, Dr. Balakishan, is a postdoctoral fellow in Japan and Canada. He spoke about all the goals, how a science student can think, how a science student can reach them. So definitely, I believe many of us got inspired with his talk. And then Dr. Ayodhya. Dr. Ayodhya is a patent officer. He is from Chennai. So he is also a son of the soil. He is Nalgonda. And he spoke about the intellectual property rights importance. Maybe it can be in the form of patent or a trademark or a license, many forms of intellectual property rights he has spoken. And also he gave a procedure how we should approach it. Generally, students or teachers may think once they do their PhD, they may go for patent. But he, in the first slide itself, he mentioned, do the patent. First go for patent, then you publish it. He also stressed upon the word called age. Age is not a matter for the patent. He gave many examples from various kids from the schools. My 4-year girl patented, and 9 years boy, 11 years boy, many school boys and girls have patented their innovation work. Maybe it's one good example. Life applications was washing machine with the help of fitness. Instead of going for gym, if you go for this cycling, if the cycling attached to the washing machine, that's also got patented. So these are the many areas which he also spoken about the importance of IPR for the students and for the teachers. What are the benefits out of this patent? Also give many informative things to follow for the teachers and students. And one more thing, the South Korean professor spoke about the internships. What are the available internships for the students and teachers? So definitely, if we have a desire, definitely we can approach, we can tap some funding so that we can do our own research. Maybe a student, a PG, from the PG student to the PhD scholar and the professor also. So these are the eminent professors which we brought, many of them from central institutes like University of Hyderabad, NIT Warangal proposed, and patent officer, and one is Telangana University. It was the combination of experienced and young officials or teachers. The first three, if you see in the banner, first three are very much experienced in their work and research. And as Mr. Gangaraju, Dr. Gangaraju, he is one of the top 100 scientists in the world. So such a candidate, such a professor has given us time. Such a professor has come into this college and delivered a lecture. Really, I must be thanking all these professors and officers who have accepted our invitation and delivered a good lecture so that we, as a teachers, our students, our scholars, can be inspired. And some of the highlighting participants' presentations was also there. Yesterday, Dr. Y. Venkateshwarlu from Tara Government College has presented his work on wealth from the waste and mimicking biotechnology issues. And Dr. Santoshi has given a lecture about the anti-cancer drugs, how she worked and she has presented. And Dr. Dayanand also spoken about anti-cancer drugs development. And Ramachandar, Dr. Ramachandar has spoken about the natural things which can be utilized for the good purposes. And Dr. Mallikarjun has spoken about the environmental science, etc. There are other scholars who have presented their abstracts. Definitely, once again, if we can go through the souvenir which we have given, definitely this will be an input. Maybe I can say this would be a take home. Maybe you might have concentrated here or not. The online presenters as well as the offline, both the speakers, the young and experienced, had conveyed their message very clearly that we must be striving to achieve these sustainable development goals. There are 17, but a science teacher or science scholar definitely can achieve 5 to 6 goals with his own work. It's not the matter of policies, but it's the matter of work that we are going to do, whether it is a village laboratory or the sophisticated university laboratory. So these are the messages that really I am thankful for the principal and the funding agencies and my team. Definitely, I'll take some other opportunity in the same talk. I want to thank my organizing team. From the team, within the team, and from the out of the team also, there are people who helped to do this. Really, I was worried till the last day. It was Sunday, so next day we had to start the conference. But Saturday up to night 4 o'clock we were two were there in the whole night was speaking and doing the things. So at 4 o'clock we finished our the compilation and everything, and we went for sleep. And next day Saturday we came to the college. But it is not just the personal effort, but definitely it was the team effort. I really thank my team for helping me on all the ways, in and out. So with these remarks, I want to conclude my convener message. Thank you all. Thank you once again for giving me this opportunity.
Thank you, Dr. Anil Kumar sir, the convener of today's national seminar. And you have worked very hard to make this seminar successfully as a convener. Now I am requesting, one person is always expecting the quality from whatever the program we had. So I am requesting Dr. YVR Prasanna Kumar sir, the Quality Assurance Cell, Internal Quality Assurance Cell, Nagarjuna Government College. Say a few words in this occasion, sir.
Good afternoon. Principal Dr. S. Upendra sir, today's chief guest Dr. Nagajyoti ma'am, Vice Principal Dr. P. Ravi Kumar, Conference Convener Dr. M. Anil Kumar, Head of the Department Zoology Dr. Josna ma'am, Head of the Department Botany Sivrani ma'am, and other dignitaries of the dais, my colleagues and students. It's a great pleasure for me to be here. At the outset, I would like to congratulate heartily the entire team, especially the convener and the co-convener of the dais, for their wonderful achievement. The beginnings are small, but when we dream, the outcome will be great. I knew from the beginning, I saw them toiling, working, running, and whatnot. They were doing everything to make it a success. And of course, the team that they had is a wonderful team. Very good choice of roles they gave to everybody. And they did the best of their efforts. They put their best of their efforts and they did the work, and really congratulate. And also I congratulate our dear principal sir for making it a grand success and timely sanctioning the things. And I am happy that the conference ended well, closed well, and with the final speech of IPR, which is one of my favorite topics. It was great to listen to the gentleman, the speaker, today's speaker. And also I believe that all the 4 SDGs were covered in this conference. All the speakers have done very well. They come prepared. They gave good, not just the abstract, but they delivered very good seminar papers here. And maybe we can take this as a beginning, and we have to go miles to go ahead. That's what Robert Frost says in one of his poems. I wish all the best to this team and the college, and to Principal Sir and others here. I wish that we have such conferences more and more in the days to come, and take our college to the zenith level. Thank you so much. Have a nice evening.
Thank you, Dr. Prasanna Kumar sir, Coordinator IQAC. Now I am requesting our college principal, Dr. S. Upendra Garu. Please say a few words in this occasion.
Good afternoon to everyone. So today's chief guest Nagajyoti Madam Garu and the dignitaries of the dais, in front of the dais, my dear student. I am very happy for the connecting the 2 days seminar in the life sciences departments. So from this department, college has the 3 major benefits. one of the first main speaker, Professor Radhakrishna sir, explains me so he is the fellow of the Science Academy. So from the Science Academy, he promised me conduct so many workshops to our college students. So previously, only we conduct this type of workshop in the only math department in the Association of Indian Science Academy, Allahabad. But sir assurance to the all the science departments, including life science, physical science, all the departments. This is the major benefit to the students. So this all of the rural background students are there. So if you see the Indian Science Academy, Allahabad, or IASC, Bangalore, or Central Universities, these workshops are there. This is a major benefit of the student. And the second one, Professor Ramachari, is my junior end from this alumni of this college. He assurance me. So every month, he gave the one extension lecture to our students. So every month, so in the chemistry. And the Nagajyoti madam is the pro vice chancellor of university, one MOU from our college and the university. This is also very, very important in the NAC point of view. So, and these are the major from college side. And also from these 2 days, so many research scholars and the speakers, so many are participated. My students is also participating. This is the encouraging of the teachers and students. This is also very, very happy. And also lastly, I congratulate all the core team and the core team, the conduct organizing team, each and everyone from college side. Congratulate the each and everyone. And the lastly, supporting team, the non-teaching staff, and each and everyone who involved in this seminar. I each and everyone congratulate. Thank you.
Thank you so much, sir. You are the backbone of the today national seminar, sir. Whenever we enter into your chamber from the beginning of the proposal up to the end of the seminar, and we give a lot of potentiality for the each and every person who make this seminar grand success, sir. Thank you so much once again. Now on the behalf of today national seminar, we are expecting the feedback from the participant. Now I am requesting Mamadi Srinivas Garu, lecturer, Department of Chemistry, say few remarks in this occasion, sir.
Very good afternoon to all the dignitaries on the dais, of the dais. So myself, Dr. M. Srinivas. So I'm working as a lecturer in chemistry in Government Polytechnic, Nagarjuna Sagar. See, I did my PhD, but I'm teaching the 8th, 9th standard chemistry to our students in the polytechnics. See, there is nothing there to teach that 8th, 9th standard students being a PhD scholar or a doctorate. But after a long time, after 9 and 10 years, I recalled all the chemistry topics. And even my PhD topic, the research topic is that photocatalytic studies of degrading the dyes. So it was very much related to the sustainable developments. So, and when I got qualified my JRF fellowship, I went to University of Hyderabad. I met T.P. Radhakrishnan sir and also Ramachari sir, and it was very great to meet again them here. And also Gangaraju sir and my own friend Ayodhya and Anil also, we are classmates in Usmania University. It is very nice to participate in this program, and I'm expecting more programs like this, and I'm waiting. Thank you for this opportunity.
Thank you, Srinivas sir. Your feedback is like yours needed to organize such a national seminar without any errors. Thank you once again, sir. Now I am expecting another participant's feedback. Now I request Mallikarjun sir, lecturer, Department of Chemistry. please say a few remarks in the today's national seminar.
Respected principal and respected chief guest and respected dignitaries on the dais and my dear students. Regarding my feedback, sir, principal sir, ఈరోజు రాగానే when I come here, it's not like seminar. ఏదో సెమినార్ అనే వరకు మనకు ఒఫిషియల్ గా వన్ బై వన్ విల్ గో లైక్ దట్ వి ఫీల్ somewhat some hurdle type like that, but it is very compatible here, very atmospherically it is very congested, I mean what you call compatible and very calm, very easy going. I did not even now I feel that it is seminar is going on like that. So very beautifully teamwork have done here, and it is what you call it, somewhat excellency is there in this teamwork. And I feel very elated to be here, sir, and I hope it should continue further in all the departments. Thank you very much.
Thank you so much, sir, for your valuable feedback. Now I am requesting today's chief guest, Dr. Nagajyoti Garu, Pro Vice Chancellor. Anybody students are there? Yes. Now I request the student participant for the feedback.
Good afternoon to one and all. My name is Kirti. I am pursuing MSc Organic Chemistry in this college. First of all, I am here to thank Professor Radhakrishna, senior faculty member from HCU. He has enlightened us with the knowledge of composites and nanoparticles. And another senior faculty member from School of Chemistry, Professor D.B. Ramachari. He has given the knowledge about advanced organic synthesis and sustainable development needed in organic synthesis, and also disseminated the knowledge about his named reactions called Ramachari reductive and alkylation and coupling. And we also learned the knowledge about recent advancements in life sciences research. At last, we had a great experience by attending this sustainable development on the role of life sciences. On behalf of this session, on behalf of the MSc Organic Chemistry, I take this wonderful opportunity to extend my heartfelt thanks to the principal and partner of the seminar. We also thank the whole organizing committee of the Today National Seminar. Thank you for giving this opportunity.
Really, your feedback is very appreciable. On behalf of the Today National Seminar, we hope, I'm requesting you, please utilize this type of national seminars in future also. Now I am requesting today's chief guest, Madam Dr. Nagajyoti Garu, Pro Vice Chancellor, West University, Andhra Pradesh. Please address the gathering, madam. And before the addressing by our chief guest, now I am requesting Dr. Anil Kumar Bajja Garu. Please introduce her for this seminar.
Good afternoon, all of you. It is my pleasure to introduce valedictory session's chief guest, Dr. K. Nagajyoti Madam. Dr. K. Nagajyoti, AICTE Margdarshak, NAC Pre-Chain Chairman and Pro Vice Chancellor of Bharatiya Engineering, Science and Technology Innovative University, has 24 years rich academic experience in various reputed academic universities and colleges across South India. Dr. Jyoti has published 31 research papers in journals and presented 46 papers in conferences. She has 3 patents to her credit, in which 2 are granted, one by Indian patents and other by Australian patents. She has received Best Innovator and Best Teacher Award for the Indian patent.
She is the recipient of many awards for academics and community services. To name a few, recent ones include Exemplary Women in Education Award by Asia Today in 2020, Best Youth Worker Award by Indian Sri Lankan Embassy in 2019, the Superwoman Award by Star Network in 2022, apart from many Best Teacher awards. Recently, she has been honored as the Best Education Leader in Hyderabad by Forever Star Group. Her long academic service as facilitator had to get recognized by AICTE as Margadarshak in 2018 to guide higher educational institutions to improve quality of education and acquire accreditation. Dr. Jyoti has been recognized as an expert committee member by Ministry of Education for NEET and SLA. With rich expertise in accreditation process of NAC, NBA, Dr. Jyoti has been guiding many colleges presently towards accreditation. As a NAC team chairman, Dr. Nalgonda-Telangana visited many universities and colleges across the country and guided many colleges for NAC and MBA accreditation. With this introduction, I'm very much happy to hand over this mic to the today's session chief guest. Thank you, madam.
Thank you, Anil Garu, for the wonderful introduction. So before I commence, actually it's not a speech. I just want to interact with you all. Okay, I'm not going to give a big talk and all. And before I start, I would like to appreciate your college principal and the conveners for organizing this event in a very successful way. I think I'm a bit late. I have come during the valedictory session. I could not listen to all other speakers, but from the feedback I understand it all went on very well and all of you gathered a lot of knowledge here. That's very good. And I have also seen the infrastructure while coming to this place. And I have visited many government colleges as NAC peer team member, but I have not seen such a good infrastructure in other government colleges, whether it is our state or other states. Wonderful infrastructure, and even I visited IQAC, and I have been telling sir, IQAC is a more challenging job. I visited the department also, and I said, as already we have signed an MOU with your college, we will be helping in various aspects like research or IQAC works, anything we will be helping. And the last 2 days, you would have heard lots about sustainability, right? So I don't want to stress much on sustainability. All of you know by now, there are 17 goals and all these are framed in 2015. Now in June, they measured how far we achieved. And can anybody say how much we have achieved India? Out of 100%, we have achieved only 65 plus. Whereas we have other countries who are reaching 80% like Sweden and Denmark. We should be very happy that we are far behind our neighbors, Pakistan and Bangladesh. But we can't be happy for that. We should reach the maximum 100. In which year we have to reach? 2030, we have to reach 100%. All these things are different. Okay. Whenever I say sustainability, okay, everybody say that yes, madam, government is taking all the steps. Modi ji is doing this, that. What are you doing is my question. As an individual, what you are doing, that is more important. When I go for NAC peer team visit, everywhere you can see logos, totally plastic banned campus. And they give us Bisleri water bottles. And again, during the exec meeting, they say it is plastic banned. But the banner is made up of plastic. It's not wrong that you use plastic, but how do you recycle? How do you reuse is very important. What do you do with these bottles? Do you know in a place, a very rural village, that is in the successful country, Sweden, there what they are doing, using these bottles, they are generating air cooling. Because
You know that whenever we use an, air cooler or AC, you are creating the greenhouse gases, right? But though we have nature, wonderful breeze outside, we all in urban areas, we close the windows, we are habituated to have air conditioners, we are habituated to refrigerators, we can't live without that. But we talk about sustainability. So what they have done is they have used the water bottles to generate the air cooling by using the advanced technology. And many women are here. I was asking actually why there is no, see, one of the goal is women equality. No. So one of the SDG is women equality. So I was asking your coordinators, why out of 8 speakers, only 1 is women? So what they told us, you are aware that is sufficient, but I want more speakers from this side also. Because whenever I go as panel chair for many of the meetings, main topic nowadays is, why is women not taking leadership roles? What women say is, ma'am, in my college, they are not giving any position, madam. All gents are taking all the top positions in private places. But reality is, nobody wants to take that because they have to work extra timings or they have to sacrifice their holidays. This is the main thing. So recently we had a meeting in JNTU where there were around 3,000 students were sitting. I asked whether any girl is ready to come forward to take up the leadership position. Not even single hand. Because we women are very happy with whatever we get. Not only women, all of you who are sitting here. What did you think is I got a government job? I settled. I am doing PhD. I am going to do PhD. And I will become a doctorate. That is fine. Okay. It's not the end. The learning has no end because nowadays, the students know much better than the teachers because they use more of ChatGPT, AI and all. If they are asking some question in the class, we will feel very embarrassing because we don't use ChatGPT and all that much. So you have to advance yourself, okay? And recently, for the past 2 days, you would have heard about various sustainability, environmental sustainability, water sustainability, energy sustainability. Have you heard of emotional sustainability? There is a word called emotional sustainability. What is that is, many people are feeling depressed, many people are feeling stressed, getting fear because of that. I'll tell you one example. So recently, 2 months back, there was a flight crash. All of you know about it and everybody dead in that, right? That was a real incident, but if you see the social media, every day, that flight collapsed, this flight crashed, they stop a big accident like this. This is emotional stress. This is, we have to make it emotionally sustainable. We have to watch the news, but what happens in social media, once you watch a particular accident, then whole Facebook you will get only about accident. Today that clash, this will make you feel emotional. This will make you feel unsustainable. So many health issues are coming. They say that 60% of the students, especially in the younger age, 13 to 20, they are getting health ailments. Even in the year of 25-30, they are getting heart attacks and strokes because of this. So we have to be emotionally sustainable. And you need not do much for getting sustainability. Enjoy the nature. You have to do hard work, no doubt in that, but take a vacation, go to a scenic place, take a break, enjoy yourself so that you will be not feeling stressed. This is one fact. And another sustainability we are talking is algorithmic stress, algorithmic sustainability. Because now everywhere AI is there, that is related to algorithmic stress. Why? Because when you search something, then AI starts showing the same thing. Okay. Once I thought of buying a very costly sofa, right? But the price was very high. I didn't want to buy it. But from the next day, solutions are coming. Same 1 lakh sofa, 2 lakh sofa. This gives me depression that I'm not able to buy that particular sofa. So this is one type of stress from where we have to come out of that. So this American Psychological Association, they are telling that social media is causing 75% of health issues in all the ages. So we have to come out of that. And since I was told that all of you are teachers here, except I think the girl who gave a very cute feedback, So I would like to tell few things to the teachers. Generally, when we do some work, whether it is for conference or anything, it can be for NAC or NBA, we feel that we are doing something for the college, not for us. So we feel more stressed, but that is not the truth. Why? Because in 2007, I started my career in 2000, but this NAC career, that is doing all the NAC work, I started in 2007. All my colleagues used to tell, why are you doing day and night all this work? Are the college paying extra money for you? No. That is the hard work I have done. Now I am the NAC peer team member. So your hard work will never go waste. You are not doing for the college, you are doing for only yourself. So always be prepared for that, and always don't be in the same region. Okay, if you belong to Telangana, always don't be happy with Telangana. Go to different places, meet different people, learn from them. You also teach them and come. Because when I go for NAC visits, I just don't go and teach them. I also learn so many things. Today I came to this college. I understood many things from here also. It's only sharing of knowledge everywhere. So in 2017, I was selected as NAC team member and AACT person, but I used to get lot of visits. I never went anywhere, apart from Hyderabad, I never traveled anywhere before 2017. Where in 2017, I joined this university, Best Innovation University, where we have our guides, we have our scholars from this college. What happened? We have a very dynamic founder of our university, Chancellor, Dr. Rupa Vasudevan. She's almost like my sister. She used to push me. We need somebody to motivate us, It may be a family member, it may be your student also. So she used to tell me, you're getting a wonderful chance to visit many places. Why you're not going? And now I covered nearly 24 peer team visits I completed in India. And I also visited many countries in the last 5 years. So almost 8 countries I have visited. But there should be some motivation. If there is nobody for motivation, you have to pat your own shoulders. So I will tell you one short story. One farmer was there. So he went to a garden where one small bird was coming out. Okay. He don't know what bird it is. He brought that cute small bird to the home. When he came home, there were so many hens and he was growing many hens. So he has put this bird also along with the hens, and this bird started growing along with the hens. Okay. But actually that small bird is eagle. It don't know its power. It is thinking I am also a hen. It lost all the power. It doesn't know to fly. Just it used to eat and roam with the hens. One day what happened? One senior elder eagle was going through that way. It saw that and got shocked. What is this? One eagle is like a hen, no power at all. So it went to the eagle and told, see ma, you are an eagle, not a hen. You please try to fly high. But this eagle is telling, no, I am only a hen. You don't know about me. I can't fly and all. Then the senior eagle takes that small bird to the mountain, trains it to fly, and then that small bird flies and flies high. So this is how you should motivate your friend should motivate you, or your family members, whatever it is. Because when we travel in our career, we can't expect everybody to motivate you. Crabs, pitalu they say. When there are so many crabs inside a basket, what will happen? If one crab is coming out, others will pull down. They won't do anything. They will not allow others also to do. Because many PhD scholars, when they want to take admission in our university, they come with their spouse. So if a lady wants to join, they come with a husband and vice versa. I see that there is a lot of support from the family members also. But even then, if nobody motivates, everybody discourages also, you should motivate yourself. So coming to the sustainability, again, there was a small fish that was swimming inside the depth of the ocean. Okay, it was asking its mother, what is water? Just imagine a fish asking its mother, what do you mean by water? All the time it is in water, but it is asking what is water. Then the mother says, even I do not know what is water. Okay, finally, one big fish, it comes and says that it takes the small fish to the surface of the ocean, and then the small fish is not able to breathe because it has come out of the water. Then immediately the big fish takes it to the ocean and says that if you don't have water, you cannot live. Many people, we have everything, but we don't realize we have all facilities. For example, if you take your college, all the facilities are there and you have seniors who can motivate you, right? But how many of you are concentrating in research? How many of you are concentrating in patents? See, all this you are doing not for the college, it's for your upliftment. Okay, so once again, I wish the college organizing committee for having such a wonderful conference. Mainly, I want to appreciate why, because this particular conference is held in collaboration with the organized PM Usha and also the Telangana Council. That is a very appreciative one. So once again, congratulations and best wishes to all the teachers. Thank you.
Thank you so much, madam, for your valuable suggestions and also explained about different type of the sustainabilities in this valedictory session. So now I'm requesting the convener, Dr. M. Anil Kumar Garu, please propose a vote of thanks for this valedictory session. Once again, good evening. So, once again, I want to propose vote of thanks to the dignitaries on the dais, especially the principal and the chief guest and the organizing team and PM Usha, Telangana Academy of Sciences, Telangana Council for Higher Education. So, in the process of proposing vote of thanks, generally vote of thanks is proposed by some other team member, but willingly I took up this responsibility because I want to thank them individually. That's why I took this. So before I go to my team, I want to thank chief guest of this function, Madam Nagajyoti Garu. So at the same time, I request a principal sir and the team, my organizing team, to honor her with the bouquet and the shawl. So my request, felicitation. No. I request Shivrani madam and Jyotsna madam to come forward and along with the principal, please. I request Vice Principal Sir and the Chief Guest and IQSC Coordinator to honor the Principal Sir. Now I request Vice Principal Sir and IQSC Coordinator and the Chief Guest to honor the Principal with the bouquet, shawl and memento. Thank you, sir, for all the support that we have received till today. Thank you very much.
I request Vice Principal, sir, come forward. I request Principal, sir, and the IQSC coordinator and my organizing team to honor the Vice Principal. Thank you, sir. Now I request the same team to honor the Quality Assurance Cell Coordinator IQSC convener to come forward and accept our honor. Thank you, sir. Definitely IQSC should be a dynamic, and you are dynamic, we believe. Continue to drive us in the same quality. I request the team and the principal IQSC and chief guest to honor zoology HOD. Dr. Jyotsna, madam. Thank you, madam, for all support. Even though you were sick in few days, but still I disturbed you even at 12 o'clock midnight also they have called you. So thank you for all the support. Now I request Shurani madam to receive the honor from the principal and the vice principal and the chief guest. Thank you, madam. All the physical arrangements, the beautiful stage you are seeing is because of the Shivrani madam and her team. So thank you very much, madam. Now my friend and co-convener, I request the principal sir, vice principal and chief guest madam to honor him with the bouquet and memento. Thank you. With no words, I'm just I'm just saying thank you to Anil. I don't have words or I cannot write all the words. The support that he gives me in every moment I take. Wherever we go, we are going together. So much, I cannot say more than this. I request one of another friend, Bhatri Nagaraju is not here due to some emergency work we have to leave Hyderabad. So I request Dr. Bhagyalakshmi madam to come forward to receive the honor. Memento and bouquet, please. Thank you, madam, for the at-home team. The shawls that you are seeing is the selection of Bhagyalakshmi madam. You cannot blame me if it is not good, but you can appreciate if they are good to Bhagyalakshmi madam. Thank you. And another my team member, Ms. Sirisha. I request Sirisha madam to come forward and receive the honor. Look and memento, please. Now I request Krishna sir, lecturer botany, to come forward and receive the honor. followed by Dr. Siddesh sir. Krishna sir is a silent worker. You can see him. He will not speak much, but he does a lot of things. And Siddesh sir also to come forward and receive the honor. Thank you, Siddesh sir. Yesterday he was at the doors. He was at the doors so that students may see the doors wide open in the world. Now I request another two, what can I say, two strict officers for the certifications. So I request Ms. Sarita madam to come forward. I got few complaints about these two young ladies saying if you are absent, you will not get any certificate. So such a young, such a wonderful team I have. Thank you very much, madam. Thank you. And I request Maheshwara madam to come forward and receive the honor. Thank you very much, madam, for your with the smile. Many participants who came, you gave a good smile, and they felt it's a home. Thank you very much. So this is the end of honoring my friends and my team. I once again thank you all for giving me this opportunity. Now, other than the team members, I have one more special friend. Whatever you do here, it is published in the domain, in the public domain. I hope you guess the person who is behind this. The PRO of this college, Dr. Nalgonda-Telangana. I request him to come forward to receive the honor. Dr. Nalgonda-Telangana. Okay. So thank you once again for helping me all these days.
We have been thanking everyone. I would like to appreciate one special person. Only through him I came to know all others, that is Munislam sir. A big applause to sir.
One more incident is a YVS sir, please. The person backbone to this one with the instructions of principal and the senior faculty members, he done everything and guided the team members in a successful manner to complete this two-day national seminar. మనిషి మందము అతని మనసు అందము ఈ కాలేజీ నుండి ఎప్పటికీ విడిపోవద్దు అతని బంధము ఈజ్ నన్ అదర్ దాన్ డాక్టర్ ఎం అనిల్ కుమార్. And a big thanks from the team members to you. Dear Anil, all the best and great success. We wish you in your future endeavors. Thank you. I am requesting all of you to stand and participate for national anthem. 1, 2, 3, start. Janagana Mana Adinayaka Jaya He Bharata Bhagya Vidata Punjab Sindhu Gujarat Maratha Dravida Utkala Vanga Vindhya Himachala Yamuna Ganga Uchala Jaladi Taranga Tava Shubha Name Gahe Tava Shubha Shisha Mage Gahe Tava Jaya Gada Jana Gana Mangala Dayaka Jaya He Bharata Bhagya Vidata Jaya He Jaya He Jaya He Jaya 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 He Jai Hind. I forgot few names, but definitely I should not forget. The technical team behind this, I want to express my heartfelt thanks to Mr. Tausif and Mr. Pravin and non-teaching staff who helped in food and many technical things. My special thanks to Pravin and Tausif. Thank you very much, both of you, for your time and energy.